Fellow sapiens, dim the lights, let your breathing slow, and let your attention drift like a small boat on a quiet sea. Tonight we will travel very far back, not in miles, but in minutes and molecules, to a sudden chill that touched the whole planet about 12,900 years ago, at a moment when people were already edging toward a new way of life. This is a calm, science-first telling. I'll hold the facts gently, admit where the map grows thin, and point clearly at evidence: the ice, the pollen, the charred grains, the DNA tucked in teeth. If you enjoy this slow, careful unfolding, please like and subscribe. It helps small channels that take time and care. Tell me in the comments, when do you listen? Late at night, early in the morning, or during a quiet commute? I place timestamps in the description for your convenience. If you want to skip ahead to methods or to a particular scene, they'll be there. Now, let's begin. Cold open. A courtyard at dawn. The courtyard is small, paved with river stones, smoothed by years of footsteps. A low, single-room house opens to it. A stone hearth still holds a bed of gray ash. Outside, a young woman draws a cloth over a shallow basket and lifts it to the light. The basket contains a few brittle seeds, small, round, pale fragments of wild emmer and wild barley, gathered the autumn before and stored with care. The air bites. Morning rises like a thin, pale sheet. Her breath forms soft ghosts, and the mundane work of the day, mending a net, scraping a grinding stone, seems edged with urgency. The river on the valley floor has slowed to a narrower pulse. The trees higher on the slope are thinner than she remembers. People do not speak much about the why. They speak about the work. Who will bring firewood? Who will tend the flocks? Which fields will be left fallow this year because the rains came late? A visiting notary, if there were such a thing in this place and time, might write a short line on a fresh skin. We saved seed. In that small sentence is the hinge on which futures rotate. If those seeds sprout, if people continue to harvest and store, the next winter will hold the possibility of building more than shelters. It will hold the possibility of choices that accumulate into societies. Far to the north, buried under many meters of ice, someone is reading a series of rings written not in wood, but in layers of snowfall. Those rings speak in the language of oxygen isotopes and trapped air, shifts in D18O, sudden jumps and dips, tiny chemical breaths that record temperature and wind, dust and melt. The ice does not know human names or ambitions. It only preserves a sequence of seasons, each one a parchment page. When scientists read that parchment, they find a passage that looks like a sentence pulled from the sky. A rapid return to much colder conditions, sustained for more than 1,000 years. The question is immediate and strangely intimate. Why exactly did the world grow colder so quickly? And why, more crucially for us, did that cold arrive at the same moment that many communities were already experimenting with intensified plant use, storage, and settlement? The very practices that, over millennia, would flower into agriculture. We might imagine a single cause, neat and dramatic. A comet that slammed and scattered dust, a pulse of fresh meltwater that stalled ocean currents, or a cascade of volcanic eruptions. Each of these ideas has been proposed, fought over, tested, and retested. But science prefers a different posture, not certainty at first glance, but patient triangulation. We read ice cores and marine cores, the chemistry in lake mud and the shapes of pollen grains, the scars in human teeth, and the broken edges of tools. Each archive is a witness with its own vocabulary. Their testimonies sometimes agree, sometimes they do not. In the courtyard, the woman presses a seed to her lip, a private, almost ceremonial gesture. She knows, in the practical way that people have known since our species first kept track of seasons, which spots on the hillside hold a little more moisture in dry years. She knows how to roast a seed to make it last a little longer. She has no sense of isotopes or ocean circulation, but she does know weather. Her choices, where to bury seed, how much to eat now, how much to save, are the quiet acts that determine whether a lineage continues. The central question that will guide us is this: when the planet shuddered into that sudden freeze, how did people respond, and how did those responses change the course of history? Did the cold slow the march to farming? Did it nudge people to store more, to settle more, to experiment more? Or did it simply rearrange already flowing currents of innovation? This series will move slowly from that courtyard to the cores drilled in Greenland and the sea, from the grain fragments in ancient middens to the isotopic chemistry buried in stalagmites. We will sit with the evidence, separate consensus from speculation, and, where appropriate, flag what remains unsettled. In the next part, we'll step into daily life before the freeze, sketching the rhythms and technologies of the Natufian and their neighbors, so that the change feels measured against what was ordinary. Dim the lights again. Let your breath fall slow and even. Picture a small cluster of stone houses pushed up against a terrace above a river valley. Smoke threads rise thin and steady from short, well-tended hearths. Dogs, small, lean, and helpful, lie coiled near doorways. Children's laughter is an occasional, cautious sound. Life here is busy with small tasks, and each task is an investment. This is the world of people archaeologists usually label Natufian in the Levant, ca. 14,500 to 11,500 years ago, a time and place when human groups experimented with new forms of settlement and foodways. They are not yet farmers in the way we later understand the word, but they are not pure nomads either. They sit on a spectrum, some seasons seeing concentrated occupation of permanent structures, other seasons involving mobility and movement. Their pattern is best described as semi-sedentary, a repeated return to favored places with enough stability to keep stored seed, to maintain house structures, and to bury their dead within the living spaces. Imagine approaching one of those houses. The walls are low and roughly circular, cobbles set with thick daubs of clay. Inside, grinding stones, flat, well-worn, bear the deep, smooth grooves of repetitive use. A shallow pit near the hearth contains a stash. Cracked stones, a string of wooden pegs, charred plant fragments. The most ordinary objects become profound in this setting. A sickle fragment, its edge worked to a sharp gloss by slicing silica-rich grasses, hints at concerted cereal harvesting. A bowl with residues of processed seeds suggests taste, effort, and choices about what to save for the lean months. The day begins with light. People bring water from near streams, but they also know where springs seep out of limestone. They choose patches of the valley floor where winter rains puddle a little longer. In these microecologies, wild cereals, emmer-type wheats, and hulled barleys grow thick along terrace edges and in disturbed soils. The knowledge we must not romanticize. It is deep, local, and learned. Knowing when a plant ripens, which slope holds more moisture, where rodents frequent, and how to keep harvested seed dry through a wet winter are practical disciplines taught across generations. A common scene, the communal processing of harvested plants. People sit in small groups around a single grinding slab, rhythmically pounding seeds into coarse meal. The sound is steady and soft, a low percussion that echoes with small talk and with the exchange of news. Beneath that sound, other work continues. Hides are worked, thin bone needles are made, hafted stone points are sharpened. Tools tell a story of mixed economies. Hunting remains important. Gazelle bones and middens are frequent. But plant resources are significant enough to require specialized gear. Storage is a crucial word. House floors often show shallow pits and built-in bins. Storage implies planning and the trust that someone will return to use what is kept. That trust, in turn, alters social relations. It creates obligations. I store for you. Next season you share your hearth. Households negotiate who keeps what, who shares labor, and how to handle shortages. These are the social experiments that precede formal agriculture, small social arrangements that scaffold the accumulation of food and thereby permit larger investments in material culture and kin networks. Diet here is mixed. Animal remains show repeated mass harvest of small ungulates and the use of wild plant foods in varying proportions across regions. We can taste this variety through residues, starch grains locked into mortars, lipid molecules adhering to pottery and stone tools, and the isotopic signatures preserved in human bone. Teeth tell intimate stories, enamelware from gritty, stone-ground meals, dental calculus preserving traces of plant microfossils, tiny chips that speak of a life using teeth as tools. Those traces are civil, blunt witnesses. They do not invent drama. They report pattern. Social life is organized by more than subsistence. Burials appear within house floors and nearby middens, an intimate geography of the living and the dead. Grave goods vary, a bead here, a shell there. Careful placement of the dead in houses suggests strong attachments to place and to particular houses as more than shelter, as anchors of history and memory. These gestures create a cultural continuity, a sense of lineage that matters when we think about how communities responded to environmental stress. People who invest in place, who anchor their dead within it, may respond to hardship differently than purely mobile groups. They have relationships, obligations, and reputations to protect. Technology is pragmatic and inventive. Microlithic stone tools, tiny, retouched blades set into composite handles, show skill in making efficient cutting edges. Sickle gloss on hafts shows repeated serial cutting. Grinding equipment is abundant, and the variety in grinding stones suggests different recipes and different textures of food. Organic tools, wooden pegs, plant fiber bindings, simple baskets rarely survive intact, yet indirect traces like post holes and impressions in ash give us hints. The material record is a palimpsest. What we see is a fraction of life, but a telling one. Seasonality shapes rhythms. Some sites show intense occupation during particular seasons, harvest times perhaps, and relative quiet in others. Migration is not absent. Herding and following flocks into new pastures remain part of life. Yet the very presence of storage, the investment in stone architecture, and the density of occupational debris suggest that many groups balanced mobility with a stronger attachment to place than earlier hunter-gatherers had. All this matters because of timing. The window in which these behaviors intensify overlaps with the climatic hiccup we call the Younger Dryas. If people were already practicing concentrated harvesting, selective storage, and place-based ritual, then a rapid multi-century cold could interact with those practices in complex ways. It could push some groups to double down on storage and experimentation. It could force others to move, to fuse with neighbors, to change hunting patterns. It might accelerate some innovations or stall others. None of this is to imply a single arc from foraging to farming. The path is messy, contingent, and regionally variable. The archaeological record shows pockets of intensification, places where cereal use rises, and other places where hunting remains central. In some sites we find clear increases in grinding equipment and storage features. In others we find continuity. The diversity itself is a kind of data. Human responses to environmental and social pressures vary across landscapes and histories, so sit with that scene a moment longer. In the courtyard, a basket of seeds rests under a cloth. A child watches an adult rub a seed between finger and thumb, testing for soundness. The household decides, quietly, today we share, tomorrow we save. Those small, repeated choices are the steady work that meets climate and chance. In the next part, we will travel from that courtyard to the coldest archives on Earth, Greenland's ice cores, and listen to the chemical lines that record abrupt change, finding the first solid cues that something large and fast was happening to the world's climate. Dim the lights and let your attention settle on a column of clear blue ice. Imagine it laid out on a laboratory bench, a long cylindrical core, the color of old glass, sliced into neat segments and labeled in a careful hand. Each slice is an archive of a single season, a stacked diary of snowfall. When scientists take a core from Greenland, they are not simply taking ice, they are taking time. Ice cores read like a layered manuscript. The most direct signal we use for past temperature is the ratio of oxygen isotopes in trapped water molecules, commonly written as D18O. Heavy oxygen, 18O, prefers warmer water and evaporates differently than light oxygen, 16O. Over long runs and careful calibration, zonal shifts in D18O map onto temperature changes above the ice sheet. When D18O dips, the simplest interpretation is that the local climate cooled. In Greenland cores, the Younger Dryas shows as a surprisingly sharp and sustained drop in D18O, a clear, crisp step down in the record. That step is striking because of its speed. The onset of the Younger Dryas appears in many Greenland cores as an abrupt change, happening within a span that is short in geological terms, years to decades. It is not a gentle slope into colder weather, it is a sharp break. The ice also contains other testimonies, layers of fine dust that increase markedly during the cold interval, indicating drier, windier conditions that carried more sediment onto the ice. Trapped atmospheric gases, tiny bubbles of ancient air, show shifts in greenhouse gas concentrations. And methane records in particular help link Greenland sequences to broader, global signals. There are multiple ice cores from Greenland that researchers commonly reference. GRIP and GISP-2, recovered in the 90s, were early high-resolution records. NGRIP extended and refined that picture. Later projects, such as NEAM, have helped clarify variations and provide better spatial coverage. Together, these cores create a relatively coherent portrait, a rapid northern cooling that persists for roughly a millennium or more, followed by an equally swift return toward warmer conditions at the end. The timing aligns, within uncertainties, with other records elsewhere on the planet in ways we will explore. How do scientists read time so precisely from these cores? The simplest method is layer counting. Each year brings a seasonal cycle: summer dust and melt, winter compacted snow. These cycles leave recognizable patterns in chemical signals and in visible stratigraphy. By counting seasonal layers, much like counting tree rings, researchers create annual chronologies. For the upper portions of Greenland cores, where layers remain discrete, this gives year-by-year resolution. For older, deeper sections, layer thinning complicates counting, and other dating methods such as synchronization via volcanic ash layers, tephra, or matching greenhouse gas spikes help anchor the timeline. There are important checks and balances in these methods. Methane, for instance, is produced by wetlands and has a global atmospheric distribution. A rapid change in methane levels recorded in Greenland ice can be matched to the same change in Antarctic cores, allowing scientists to synchronize the two records and get a sense of global timing. Volcanic ash grains found in ice provide distinct chemical fingerprints that can be linked to known eruptions and independent radiocarbon-dated terrestrial records. These cross-checks increase confidence in the chronology even as each archive has its own quirks. The ice itself records more than temperature. The chemistry of trapped aerosols, sulfate, nitrate, and a host of trace elements speaks about volcanic inputs, biomass burning, and dust loads. During the Younger Dryas, Greenland ice shows increases in dust and changes in aerosol composition consistent with colder, drier conditions in regions that supplied windblown particles. That change in dust also tells us something about the hydrological cycle. Less moisture on the landscape, more exposed sediment to be lofted and transported. Ice cores are also where the idea of abruptness took hold. Before these records, climate change was often imagined as gradual, unfolding over millennia. The layered precision in ice cores taught us that significant, sustained shifts can happen within lifetimes. That lesson reframes how we imagine human responses. Communities living through the Younger Dryas may have experienced climate shifts within a few generations, not over the slow sweep of deep time. Yet ice cores are not the final word on mechanisms. They give us a clear, high-resolution sense of what happened in the Greenland region, and, through gas synchronization and global markers, they help map broader timing. What they do not, on their own, fully explain is the cause. Did a sudden pulse of meltwater from proglacial lakes alter ocean circulation and trigger northern cooling? Did volcanic aerosols or cometary dust dim the sun? Or did internal variability within the ocean atmosphere system do it without a single external trigger? The ice points to an event and constrains when it occurred. Explaining why requires bringing in other archives: marine cores, lake sediments, terrestrial pollen, and models that test how the climate system responds to different forcings. We should also be candid about uncertainties in ice records. Layer counting becomes more challenging where melt layers obscure seasonality. Post-depositional processes can alter chemical signals. Gas age-ice age differences, the fact that air trapped in ice may be younger than the surrounding ice due to diffusion in the firn layer, require correction. Scientists use sophisticated models to account for these effects, but the corrections introduce modest uncertainties. Even so, the overall pattern of abrupt northern cooling during the Younger Dryas is robust across multiple Greenland cores and corroborated by complementary records. Imagine then a person peering at the ice core in a quiet lab, the subtle banding like frozen grain, the thin dust horizons, the tagged labels. Each thin band whispers of a winter wind, a volcanic puff, a season of grain. When those whispers stack into a long, sharp fall in D18O, we feel the scale of change in a new way, not as a rumor, but as a measurable, synchronous signal across one of Earth's key climate archives. In the next part, we will follow the water, literal pulses of fresh meltwater, and the ocean currents that redistribute heat. We'll ask how changes in the North Atlantic circulation might have amplified or even initiated the Younger Dryas, and we'll listen to marine cores as they tell their own complementary story. Dim the lights. Picture the ocean as a slow, patient engine, a vast conveyor belt of warmth and salt that nudges climate in distant places. At the heart of that engine is the Atlantic, where warm surface waters travel north, give up heat to the atmosphere, grow dense, and sink. That sinking pulse helps pull the rest of the ocean along. Scientists give this system a name, the Atlantic Meridional Overturning Circulation, or AMOC. It sounds technical, but the idea is simple. The ocean moves heat around the planet, and small changes in its salinity or temperature can shift where and how that heat is delivered. Imagine rivers not only carrying water across land, but also carrying signals into the sea. Meltwater from ice sheets is fresh and light. If a very large pulse of fresh water enters the North Atlantic, it can lower surface salinity there. Fresh surface water resists sinking. Colder, saltier water sinks more readily. In some climate models, a big enough freshening can slow or even briefly stall the overturning circulation. The immediate consequence is less heat transported northwards, and regions that used to be warmed by the ocean grow colder. That mechanism is one plausible bridge between continental melting and the sharp northern cooling seen in Greenland ice. To read how the ocean responded, scientists drill marine sediment cores. These long tubes of mud and sand, recovered from the seafloor, are time capsules. Each layer traps the biological and mineral debris of its era, shells of tiny plankton, chemical signatures, and sometimes even little pebbles dropped by icebergs. Among the most useful witnesses are foraminifera, microscopic shelled organisms that live in different water conditions. Their shells record oxygen isotope ratios and trace element chemistry, and the assemblages of species tell us about past temperatures and water masses. When surface waters cool or freshen, the community of forams shifts in predictable ways. One proxy often used is the D18O of foraminiferal calcite. This signal blends temperature and the isotopic composition of seawater, which itself is influenced by salinity. To tease these influences apart, researchers pair D18O with independent temperature proxies. For example, AGH-Ca ratios in foraminifera or alkinone unsaturation indices from certain algae. By combining proxies, they can estimate past sea surface temperatures and infer changes in salinity. Another powerful line of evidence comes from ice-rafted debris, small stones and pebbles that icebergs carry from continental margins into the ocean and drop as they melt. Layers rich in IRD mark intervals when icebergs were calving and traveling south, offering a visible fingerprint of glacial instability. What do these marine records say about the Younger Dryas? In the North Atlantic, many cores record changes consistent with colder sea surface temperatures and increased iceberg activity close to the onset of the event. Some sites show sharp changes in species composition and sediment characteristics, suggesting rapid shifts in the local marine environment. Around the same time, proxies for freshwater input, changes in D18O of seawater inferred by proxy combinations, shifts in isotopes that hint at continental water sources, and layers of fine-grained sediments carried by meltwater plumes sometimes show pulses that could be interpreted as increased discharge from melting ice. A prominent narrative ties the Younger Dryas to a massive outburst from proglacial Lake Agassiz in North America. The simplest version runs, as ice sheets retreated, meltwater pooled to form Great Lakes. At some point, one of these lakes breached its ice or morainic dam, releasing a large volume of fresh water into the North Atlantic through river systems. That fresh water, the story goes, freshened the surface North Atlantic enough to slow the overturning circulation, reducing northward heat transport and initiating abrupt cooling. The timing of that proposed outburst aligns, in broad strokes, with the Younger Dryas' onset in some chronologies, which is why the idea has been influential. But science is rarely satisfied with a single, tidy explanation. Marine records are spatially variable. The magnitude and timing of freshwater signals differ by core and by basin. Some sites show strong evidence for freshwater input, others do not. The dynamics of routing matter, whether meltwater reached the Labrador Sea, the Erminger Sea, or was diverted via the St. Lawrence influences how effectively it might alter deep water formation. Moreover, the ocean system has internal thresholds. In some model simulations, even modest freshwater injections into a sensitive state can trigger a larger circulation response. In other simulations, larger inputs are required. The truth may be a combination, pulses of fresh water interacting with a system already primed by background cooling. Dating marine cores introduces its own complications. Marine radiocarbon is subject to reservoir age effects. The apparent age of surface waters can differ from atmospheric age because of mixing and upwelling. These offsets require careful correction and sometimes introduce uncertainties of centuries, significant when debating abrupt events. That is why researchers often seek multiple lines of synchronization: tephra layers, volcanic ash with a geochemical fingerprint, abrupt methane shifts tied to wetland emissions visible in ice cores, and stratigraphic matches across cores. There are also insights from the wider ocean. The Younger Dryas is not purely a northern story. Changes in the tropical and southern oceans likely contributed to and responded to the perturbation. Marine cores from farther afield record temperature and circulation changes that help place the event in a global context. The ocean is a connected system. A change in the North Atlantic can propagate through wind patterns, sea ice expansion, and teleconnections to influence rainfall and seasonality in regions as far away as the Mediterranean and the Near East. Importantly, marine evidence paints a nuanced picture. While freshwater forcing is plausible and supported by some proxy records, it is not an uncontested universal signal in every core. Some researchers point to evidence of increased iceberg calving and regional cooling without a clear, singular freshwater pulse. Others highlight synchronous changes in atmospheric circulation or in terrestrial records that suggest a more complex interplay of drivers. For our purposes as listeners, the ocean's response is a reminder of scale and connectivity. A pulse of meltwater on a continental margin is not a local curiosity. It can, under the right conditions, shift the engine that moves heat across hemispheres. That engine's slowdown reverberates in seasons, in growing seasons, and in river flows. For the farming question, for communities gathering seed and making decisions about storage and settlement, those marine shifts are not distant abstractions. They help set the rhythm of rains, the abundance of wild cereals, and the reliability of river-fed wetlands that supported both people and the plants they used. In the next part, we'll return to the people, how Natufian and neighboring communities show archaeological traces of coping and change, and which material signatures archaeologists read as resilience, stress, or transformation. Dim the lights and lean in a little closer to the small things: a stone ground smoothed by centuries of grain, a pit lined with cobbles and ash, the whisper of a seed crushed to flower. Archaeology's strength is its patient attention to such details. From them we reconstruct how people lived, how they stored, and how they adapted when weather and seasons shifted. Begin with houses. In many Levantine sites often associated with late Pleistocene and early Holocene foragers, we find durable, built structures: stone foundations, post holes, and carefully prepared floors. The architecture matters because it signals investment in place. When people build substantial houses, they commit to revisiting and maintaining sites across seasons and years. That commitment is visible in the layers of debris inside dwellings, repeated floors, hearth repairs, and accumulations of domestic refuse. During times of stress, such investments can either create vulnerability or enable resilience. A fixed home makes it easier to keep stored food dry and secure. It also ties people to a location that may become less tenable if the environment shifts dramatically. Storage features are particularly revealing. Archaeologists find pits dug into house floors, or adjacent courtyards, sometimes lined with stone or plaster, used to hold seed and processed food. The presence of storage implies choices about risk management: save for drought, share with kin, or trade for other goods. At several well-studied sites, the density of grinding stones and the architecture of storage increase in layers dated to periods that roughly overlap the Younger Dryas window. More grinding stones and more storage capacity suggest intensified plant processing and a greater emphasis on buffering uncertain seasons. Toolkits shift too. Sickle blades with characteristic gloss from cutting silica-rich grasses appear in greater frequencies in some sequences. Microliths and composite tool technology persist, but there is often more evidence for plant-focused implements: mortars and pestles, querns, and tools suited to harvesting and processing small-stemmed cereals. These technological shifts may indicate that communities were investing more labor into extracting calories from plants that had long been part of the diet, rather than abandoning plant use in favor of purely hunting-based strategies. Dietary signals are subtle and powerful. Macrobotanical remains, charred grain fragments recovered from hearths and middens, document which species were collected and consumed. In some localities, charred wild cereal remains increase in abundance in layers contemporaneous with early Younger Dryas dates. Microfossils preserved in dental calculus, the tartar on ancient teeth, and on stone tools, starch grains and phytoliths, give another window into plant use. Stable isotope analysis of human and faunal bones reveals changes in the relative contribution of plant versus animal protein in diets, and shifts in local water and vegetation regimes. Taken together, these lines of evidence often point to a greater reliance on plant foods in certain regions and sites during this interval, though the pattern is not universal. Social practice shows through mortuary behavior and household layout. At some sites, the intensity of burial use and the placement of graves within living areas continue across the climatic transition, suggesting a persistence of place-anchored social continuity. Elsewhere, abrupt changes in burial custom, decreases in site density, or layers of abandonment hint at stress and movement. The archaeological record therefore shows a mosaic, pockets of continuity and intensification, interleaved with episodes of abandonment or reduced occupation intensity. This patchwork response is important. Different communities, with distinct histories and resources, made different choices under similar broad climatic pressures. Another signal is animal remains. In many stratigraphic sequences, the composition of hunted species shifts. Some sites show a move towards smaller, more easily procured prey, or greater reliance on animals adapted to open, colder landscapes. In other locations, the relative abundance of herdable species declines or becomes more variable. These faunal changes can reflect local ecological shifts, vegetation zones moving downslope, wetlands shrinking, and they can also reflect human adaptation, targeting different species or intensifying gathering when big game becomes scarcer. Behavioral flexibility is visible in subtle practices as well. Evidence for increased food processing, more grinding, changes in seasonality of occupation, shorter or longer stays, and expanded plant use breadth, including edible roots and wetland plants, are all signs of adaptation. Exchange networks might shift too. Obsidian sources, marine shell imports, or exotic beads appearing or disappearing in assemblages suggest changes in mobility and social ties. In times of stress, communities may widen exchange to secure resources, or they may collapse into more local, insular networks depending on circumstances. Importantly, the archaeological data also show technological conservatism alongside innovation. Many microlithic traditions persist through the early Holocene. People did not abandon what worked. Instead, they layered new practices over old ones, increased storage and processing sitting beside long-used hunting technologies. This blending suggests an incremental, opportunistic approach. Invest where it makes sense, conserve where necessary. Interpreting stress versus resilience demands care. A rise in stored grains could mean scarcity, hoarding against hard winters, or it could mean surplus and the social ability to set aside food for future use. A decline in site numbers could indicate migration away from marginal zones, or a reorganization into fewer but more densely occupied centers. Archaeologists weigh context. Are storage pits full of moldy, discarded grain, or are they well constructed and dry? Do abandoned houses show signs of haste and burning, or do they appear carefully vacated? These fine distinctions matter. We must also be honest about the limits of the record. Preservation biases shape what we see. Organic baskets and perishable bindings seldom survive, and the archaeological signature of short-term camps is faint. Radiocarbon dating comes with uncertainties on the order of decades to centuries that can complicate precise correlations with abrupt climate markers. Yet even within these limits, convergent signals, more grinding stones, more storage features, shifts in faunal remains, build a persuasive case that many communities adjusted their subsistence and settlement strategies during this interval. Finally, the human side. Adaptation was, and always is, a social process. Choices about what to harvest, whom to trust with stored food, when to move, and when to stay are made in households and across kin networks. They are negotiated through custom, ritual, and sometimes conflict. The archaeological markers of those negotiations, hearth repairs, shared grinding slabs, communal structures, suggest that responses to the Younger Dryas were rarely solitary acts. They were communally embedded decisions. In the next part, we'll step into a different register. We will examine prominent myths and sensational claims, the comet hypothesis among them. and begin to separate cinematic stories from the testable evidence that scientists use to evaluate them. Dim the lights again. Breathe slowly. In this part, we sit with the tools that let us turn fragments of the past into dates and sequences we can trust. The story of the Younger Dryas depends on careful timing. When exactly did things change, and how closely do different archives line up? To answer that, scientists use a toolbox of chronological methods, each with strengths, limits, and specific assumptions. Here are the main ones you'll meet as we move through the evidence. Radiocarbon dating. The familiar clock, radiocarbon dating, rests on a simple biological fact. Living things exchange carbon with the atmosphere. A steady trickle of cosmic rays produces radioactive carbon-14 in the upper atmosphere, which oxidizes to CO2 and mixes into the carbon cycle. Plants take it up. Animals eat the plants. While an organism lives, its ratio of 14C to stable carbon is roughly in equilibrium with the atmosphere. When it dies, input stops and 14C decays away with a characteristic half-life, about 5,000 years. Measuring the remaining 14C gives a radiocarbon age, the time since death in radiocarbon years. A few important practical points. Modern labs use accelerator mass spectrometry, AMS, which measures 14C atoms directly and requires much smaller samples than older methods. Pre-treatment in the lab removes contaminants, humic acids, conserved organics, that could skew ages. But raw radiocarbon years are not calendar years. Atmospheric 14C production varies through time due to solar activity and Earth system changes. So, scientists apply calibration curves to translate radiocarbon years into calibrated calendar ages. That calibration introduces wiggles and plateaus. Sometimes a range of radiocarbon ages corresponds to a relatively broad span of calendar years, which reduces precision. Marine and reservoir effects complicate things further. Organisms growing in oceans, lakes, or certain springs incorporate carbon from sources that may be centuries older than the atmosphere. That reservoir age makes marine samples appear artificially old unless corrected. Local reservoir offsets, called DR in specialist parlance, vary by region and must be measured or estimated. For the Younger Dryas, where terrestrial and marine archives both matter, careful application of reservoir corrections is essential. Uranium-thorium dating. A complementary clock, uranium-thorium youth dating is built around a different decay system and different materials. Uranium dissolved in water can be incorporated into carbonate deposits. Speleothems, stalagmites, stalactites, corals, and certain lake carbonates are typical targets. Uranium isotopes decay to thorium. Because thorium is insoluble in water, a carbonate that formed with dissolved uranium will have essentially no initial thorium, and the growth of 230th over time records age. UTH can produce very precise ages, often with measured uncertainties of decades, to a few years in well-behaved samples, and it extends beyond the practical limits of radiocarbon. Its strengths make it invaluable for building high-resolution terrestrial chronologies, especially where speleothems preserve continuous growth through key intervals. However, UT assumes the system remained closed to uranium and thorium movement since formation. Diagenesis, or later fluid percolations, can alter isotope ratios, so labs screen samples carefully and use geochemical tests to detect open system behavior. Stratigraphy and layer counting. Reading the order of events, stratigraphy is the simplest logic. Younger layers sit above older ones unless disturbed. That basic ordering, combined with contextual information about human activity and sedimentation, gives a relative timeline that radiometric dates can anchor. Involved lake sediments, annual laminations, light and dark couplets representing seasonal deposition, can be counted like tree rings to produce high-resolution annual chronologies for centuries to millennia. Similarly, ice cores preserve seasonal layering that permits year-by-year counts in many sections. Layer counting provides a direct way to measure rapidness. If you can count layers through a transition, you can bound the duration in years. But counting requires clear, undisturbed layering. Melt layers, bioturbation, or compression can obscure the annual signal and introduce uncertainty. That is why multiple independent dating methods are often combined. Tephrochronology, volcanic fingerprints. Volcanic ash layers, tephra, are powerful tie points. Each eruption produces glass shards and mineral crystals with a geochemical fingerprint. If the same tephra appears in an ice core, a marine core, and a terrestrial sequence, and if that eruption can be independently dated, then those archives can be synchronized. Geochemical analysis of glass shards provides a barcode to match layers across wide regions. Tephra is particularly valuable when it sits between or within dated levels. It constrains relative order and, when well characterized, narrows absolute age ranges. Bayesian chronological modeling. Combining evidence intelligently, modern chronology often uses Bayesian statistical frameworks to integrate different dates and stratigraphic constraints. Imagine you have several radiocarbon dates from different depths, a known order of layers, and an independently UT-dated speleothem nearby. Bayesian models treat dates probabilistically, enforce ordering rules, this layer is older than that one, and generate posterior age estimates that are often narrower than the raw inputs. The method explicitly incorporates uncertainties and yields probability distributions for event ages rather than single-point estimates. It's a principled way to make the best use of messy, real-world data. Key laboratory concerns and pitfalls. Dating is only as good as sampling and pre-treatment. Contamination with modern carbon, old carbon, e.g. reworked charcoal, or carbonate leaching can bias ages. For radiocarbon, selecting short-lived single-entity samples, seeds, leaves, or single twigs, reduces inbuilt age problems that come with wood or long-lived charcoal. For youth, careful sampling away from visible alteration and screening for open system indicators is essential. Labs report uncertainties, but understanding what those numbers mean, the confidence interval around an age, is crucial for interpreting the timing of rapid events. Ethical and practical notes on destructive sampling. High-resolution dating often requires taking tiny, destructive samples from valuable archives. A sliver of a speleothem, a charcoal piece from an archaeological feature, a thin section of a bone. Scientists now balance the need for data against the duty to preserve cultural heritage. Many projects consult with local communities and heritage authorities, minimize sample sizes using micro-drilling and AMS, and document exactly where material came from. Ethical sampling protocols and transparent data sharing are now standard practice in reputable projects. They protect both science and the people whose histories we study. Cross-checking and concordance. Why multiple methods matter. No single method stands alone. Radiocarbon, UT-VARV counts, tephra, and ice core layer counting each have strengths and blind spots. When multiple independent archives and dating techniques point to the same timing within their uncertainties, our confidence grows. Where they disagree, those mismatches are not mere annoyances. They are clues. They push scientists to refine calibration curves, to reassess reservoir corrections, or to question assumptions about open system behavior. The dialogue among methods is where chronological progress happens. In the next part, we will read the microscopic record of plants, pollen, spores, and plant fragments, and learn how vegetation changes map onto seasonal shifts and human opportunity. That pollen record will show us how landscapes changed as the Younger Dryas cooled the world, and what that meant for the people who gathered, seed-intended hearths. Dim the lights and breathe in slowly. Imagine leaning over a quiet lake at dawn, the water glass smooth, a reed trembling at the edge. Now imagine lifting a long, dark cylinder of mud from that lake, a core whose layers are like pages in a slow book. Each page contains fragments of plants, microscopic pollen grains, tiny spores, and bits of leaf and charcoal, all sunk where they fell. Those grains are small but eloquent witnesses. They tell us which plants dominated the landscape at different moments in time. Paleontology is the study of those pollen grains. Under a microscope, specialists can distinguish the shape, size, and ornamentation of pollen produced by trees, shrubs, grasses, sedges, and wild cereals. Because many plants produce large amounts of distinctive pollen, and because lakes and bogs collect that pollen from surrounding landscapes, lake sediment cores give a spatially integrated signal of vegetation change. The proportion of tree pollen versus herb pollen, the rise and fall of cereal-type pollen, and the appearance of indicator species all help reconstruct past climates and habitats. One of the clearest broad signals in pollen records across parts of the northern hemisphere during the Younger Dryas is a shift from woodland-dominated assemblages to more open herb and steppe-dominated ones. In the Fertile Crescent and Mediterranean margins, this often looks like a decrease in pollen from oak, pistachio, and other woodland species, accompanied by increases in artemisia, wormwood, Chanopodiaceae, saltbush-type plants, and steppe grasses. That pattern implies cooler, drier conditions and a contraction of forested patches. Reduced tree cover changes not only the visual landscape, but also soil moisture, runoff, and the microhabitats where wild cereals and wetland plants grow. But the pollen record is spatially heterogeneous. A lake in a sheltered valley might preserve more arboreal pollen because trees persist nearby, while a steppe margin pond will show stronger herb signals. For people living in the Fertile Crescent, those differences matter deeply. Wild cereal stands, populations of emmer, einkorn, and hold barley, prefer certain microhabitats. Disturbed soils on terraces, river margins, and the edges of fields where moisture collects. If a regional pollen record shows a net decline in favorable habitats, local patches could still persist where microclimate and soil conditions allowed. In other words, widespread cooling does not instantly erase every favorable nook. It reshapes the map of opportunity. Palynologists also look for the smaller signals that speak directly to human plant use. Charred cereal grains and phytoliths, silica bodies that preserve the shape of plant parts, in archaeological middens are the most direct evidence for plant exploitation. In some sequences, grains of wild barley and wild wheat increase in abundance in layers dating to the Younger Dryas interval. That rise could reflect intensified gathering, where wild stands became more attractive relative to falling big game availability. Alternatively, it could reflect greater burning and clearance that increased pioneer habitats for cereals. Distinguishing these causal pathways requires pairing pollen with archaeological, charcoal, and sedimentary evidence. Another set of plant clues comes from wetland taxa. Many reliable water-loving plants, reeds, sedges, and certain rushes, serve as indicators of local wet conditions. In some lake cores, a decrease in sedge and reed pollen during the Younger Dryas suggests shrinking wetlands or seasonal drying. For communities that relied on riverine and marsh resources, which include both edible plants and animals attracted to wetlands, such contraction would have practical consequences. Wetlands concentrate resources. They offer tubers, rushes for thatching and mat making, reeds for baskets, and game that visits water. Losing them alters dietary options and material supply chains. The pollen record can also detect fire regimes. Charcoal particles preserved in sediment layers vary in size and amount. Peaks often indicate episodes of burning at the landscape scale. Increased charcoal can mean more frequent human-set fires for clearance or warmth, or it can mean larger natural fires promoted by drier conditions and wind. When charcoal peaks align with increases in pioneer species and cereal pollen, the composite signal can suggest human landscape modification, intentional burning to encourage plant stands. Again, interpretation is contextual and cautious. Correlation does not equal intent unless supported by archaeological context. Precision and timing matters. High-resolution cores with varves or well-constrained radiocarbon ages allow a closer match between pollen shifts and the onset of the Younger Dryas, as recorded in ice cores. In many well-dated sites, the shift to more open vegetation types lines up with the rapid cooling in Greenland within the uncertainties of dating. Where the chronology is sharp, that synchrony strengthens the inference that climate forcing shaped vegetation patterns and thus resource availability for human communities. We should be careful with a common pitfall. Pollen percentages are relative. A decline in tree pollen percentage does not always mean fewer trees in absolute terms. It can also mean an increase in herb pollen that changes the proportions. Absolute concentrations, when calculated, offer clearer indications of true increases or declines. Palynologists therefore often report both percentages and concentration values, and they examine sedimentation rates to understand whether observed changes reflect local ecological shifts or broader regional trends. Uncertainties persist. Pollen transport distances vary by taxa. Wind-pollinated plants, like many trees and grasses, disperse pollen widely, while insect-pollinated plants remain local. Taphonomic processes, how pollen is preserved, differ by site. Human impacts can be subtle and uneven, and separating climatic drivers from anthropogenic ones is a major interpretive challenge. Nevertheless, the convergent pattern of forest contraction, steppe expansion, wetland reduction, and changes in charcoal and cereal indicators across many records paints a coherent picture. The Younger Dryas brought cooler, generally drier conditions that reshaped the landscapes on which people relied. For foraging communities, those landscape changes matter at the scale of footsteps. Imagine a winter when the reed beds that usually provide rushes and edible tubers thin at the margins. Imagine summer pastures that dry sooner, forcing herders to move earlier and farther. Imagine small patches of wild emmer that once grew in abundance on terrace edges, shrinking into scattered stands. Each of these shifts recalibrates decisions: when to harvest, how much to store, when to move, and whether to invest labor into encouraging particular plant communities. Listen to that reed by the lakeside again. The mud core holds its tiny grain, and in the lab, under high magnification, a paleontologist counts the spirals of plant testimony. The rise and fall of taxa are slow, readable pulses. They are not narrative drama, but evidence of living systems responding to changed seasons. In the next part, we will turn to human bodies themselves, what teeth, bones, and ancient proteins can tell us about diet, stress, and movement during this time. We will look for the biochemical signatures of changing meals and the lines in bone that mark childhood stress. Dim the lights, settle into your breath. We left the reeds by the lakeside and the pollen in the mud. Now imagine a small, quiet bone box on a lab bench. Inside are fragments that once held a living heart, teeth, a femur, tiny bits of skull. These remains carry biochemical stories, the fingerprints of meals, weaning, movement, and childhood hardship. They speak in isotopes, proteins, microscopic grit, and the slow curves of bone growth. Begin with teeth. Tooth enamel is hard and durable. It forms in childhood and then stops. That permanence makes enamel an archive of early life. When scientists sample enamel, or the dentine beneath it, they measure stable isotopes of carbon and nitrogen, or, more precisely for teeth, sometimes dentine collagen or carbonate. The ratios of carbon isotopes, commonly noted as D13C, tell us about the types of plants eaten directly or eaten by the animals we consumed. C3 plants, temperate grasses, and most wild cereals have different D13C values than C4 plants, tropical grasses, some sedges. In the fertile crescent of the Younger Dryas, most wild cereals are C3, so shifts toward less negative D13C might signal increased consumption of other resources or marine influence, depending on context. Nitrogen isotopes, D15N, speak to trophic level. Higher D15N usually implies more animal protein in the diet, meat or aquatic resources, while lower values suggest more plant-focused diets. When we measure bone collagen for D13C and D15N, we get a long-term average signal of adult diet over the last years of life. Dentine in teeth can be sampled incrementally to reveal childhood diets. The weaning process shows as an initial high D15N when an infant metabolizes breast milk, and a decline as solid foods replace milk. For the Younger Dryas, dentine records sometimes show earlier or more prolonged weaning in certain regions, a subtle hint that social and environmental stress influence childhood feeding practices. Beyond carbon and nitrogen, strontium isotopes, 87 Surexer, and oxygen isotopes, D18O, in tooth enamel help reconstruct movement. Strontium ratios reflect local geology. Plants and water take up strontium from soils. People who grow up in one geological catchment, snow, carry that signature in their teeth. If an adult's bone strontium differs markedly from their tooth enamel, it suggests mobility between youth and adulthood. Oxygen isotopes record aspects of drinking water and climate. Together with strontium, they map childhood landscape and later life movement. These markers help archaeologists ask who moved, who married in from elsewhere, and how far social networks stretched. Now listen to the quieter, molecular voices, ancient DNA and proteins. Ancient DNA, aDNA, from bone and tooth dentine can tell us ancestry, kin relationships, and, in some cases, dietary adaptations or pathogen presence. Preservation is fickle. Cold, dry environments preserve DNA better than hot, wet ones. Where DNA survives, genome-wide analyses can show population continuity or influx, demographic contractions or expansions, and links across regions. For the Younger Dryas window, aDNA studies have illuminated patterns of regional continuity in some places, and genetic mixing in others. But remember, DNA is a powerful tool and also limited by preservation and sampling bias. A single recovered genome is one data point in a noisy map. Proteomics, the study of ancient proteins, is increasingly important. Proteins can survive where DNA does not, especially in warmer climes. Dental calculus, the hardened plaque on teeth, is a remarkable preservative. It traps food microremains, starch grains, phytoliths, smoke particles, and proteins from plants and animals. Proteomic analysis of calculus can reveal molecular traces, milk proteins, plant storage proteins, or animal muscle proteins. For early contexts, calculus has demonstrated direct evidence of plant processing and occasional animal products. Proteins are less easily contaminated than some organic residues, but their identification requires careful lab work and cautious interpretation. Bone chemistry records physiological stress as well. Lines in teeth, known as enamel hypoplasia, form when childhood growth is interrupted by malnutrition or disease. Harris lines in long bones, visible in radiographs, mark episodes of growth arrest. Elevated levels of certain trace elements, lead or others, can indicate environmental exposure, but they are tricky to interpret archaeologically. Skeletal markers of activity, such as enthesial changes where muscles attach to bone, offer clues about habitual labor, more grinding, more carrying, different patterns for men and women in some societies. Together, these indicators sketch a picture of daily life and biological strain. Is there a consistent signal across human remains from the Younger Dryas? The answer is regionally nuanced. In many Levantine sequences, stable isotopes from bone collagen show a mixed diet, an ongoing combination of meat and plant resources. In some localities where archaeobotanical and pollen evidence points toward intensified cereal use, isotopic shifts align with increased plant contribution. In others, faunal signals suggest continued reliance on hunting. Incremental tooth analyses sometimes reveal childhood stress episodes, clustered in time windows that overlap climatic stress, suggesting increased biological strain in some populations. But this pattern is not universal. Some communities show resilience with little clear increase in stress markers. There are provocative microevidences too. Starch grains and phytoliths, trapped in dental calculus and on stone tools, provide direct proof of plant consumption and processing. Charred grains in hearths confirm which species were cooked. When proteomics retrieves plant storage proteins linked to cereals, it strengthens the inference that people were not only gathering, but processing cereals in ways that left molecular traces. Each line of evidence is a corroboration, and together they make a stronger case than any single method alone. We must pause for an ethical note. Human remains are not just data, they are ancestors and members of communities. Sampling for aDNA or proteomics is destructive and often irreversible. Good practice requires consultation with descendant communities or local stakeholders where appropriate, minimal sampling strategies, transparent reporting, and ethical oversight. Many modern projects follow these protocols rigorously. Respect for cultural heritage matters as much as scientific curiosity. Finally, temper certainty with humility. Preservation biases mean we sample a non-random subset of the past. Hotter regions often yield poorer DNA. Small bones may be absent from sites. Some social groups leave little skeletal trace. The biochemical archives are powerful but imperfect. They are conversations, not proclamations. In the next part, we will return to myth and spectacle, the comet and black mat claims, and examine how researchers test dramatic ideas with careful stratigraphy and multiple proxies. Dim the lights. Let your breath slow as you let a single thought settle. Extraordinary claims require extraordinary evidence. Tonight we will sit with one of the most dramatic stories ever told about the Younger Dryas, a story whose images are cinematic. A sky of fragments, sudden fires, a thin, dark layer across continents. It is a tempting narrative. It is also one that science must test, patiently and carefully. Begin with the image that captures attention. A thin, dark, organic-rich horizon cropping out in lakes and marshes across parts of North America and some other regions. Archaeologists and sedimentologists often call it a black mat. In some contexts, this layer sits close in time to the onset of the Younger Dryas and overlies cultural horizons used by people then. Early in the 21st century, a group of researchers proposed a bold interpretation. The black mat, along with microscopic spherules, elevated platinum group elements, nanodiamonds, and charred and melted-looking materials were residues of an extraterrestrial impact or airburst that struck the Earth around 1200 years ago. The hypothesis promised a single, dramatic trigger for abrupt cooling and, for some, a partial explanation for megafaunal declines or cultural shifts. That hypothesis is a useful case study in how science separates plausible story from tested inference. The initial claim bundled several lines of evidence together: exotic particles and sediments, peaks in certain elements, layers of carbonized material, and coincident cultural and ecological signals. The rhetorical power of a comet striking Earth is obvious. It makes the past feel immediate and catastrophic in ways that are easy to visualize. But the power of an idea does not substitute for reproducible, stratigraphically secure data. So, let us examine the components calmly. First, the black mat itself. Black organic layers are not unique to one cause. They form when organic matter accumulates in waterlogged conditions, when a marsh becomes more anoxic, when sedimentation slows and plant decay concentrates. Such layers can result from rising groundwater, changing drainage, increased organic productivity, or human activity such as prolonged habitation and associated waste. In short, a black mat is a sign that something in the local environment changed. It is not a unique fingerprint of impact. Interpreting it requires careful local context. Is the organic layer regionally synchronous? Does it contain evidence of burning? Are there abrupt erosional contacts above or below? Each site must be read on its own terms. Second, microspherules and alleged impact markers. Small rounded spherules rich in iron or other elements attracted attention because they can form in high temperature events. But they also form in many terrestrial contexts: volcanic eruptions, industrial processes, certain types of wildfire combustion, and in some cases even from diagenetic alteration of sediments. The reproducibility of such spherule finds has been a central sticking point. Multiple independent teams have attempted to sample comparable horizons and either failed to replicate the same abundance of exotic particles or found alternative mineralogical explanations for them. Science leans on replicability. If a purported global layer contains a distinctive particle type, that particle should be detectable in well-correlated cores across broad regions, under consistent sampling protocols. Third, nanodiamonds and platinum anomalies. Nanodiamonds, tiny carbon structures that can form under high pressure, high temperature conditions, are evocative evidence if they are reliably above background and properly identified. But their identification requires rigorous techniques. Contamination, laboratory artifacts, and misidentification of other carbon forms complicate claims. Similarly, spikes in platinum group elements can indicate an extraterrestrial input, but they can also be produced by localized geochemical processes or by sampling bias. In many cases where such anomalies were first reported, follow-up studies found either weaker signals or ambiguous contexts. Fourth, burned bones, fire bones, and charcoal. Layers rich in charcoal and thermally altered bone can indicate large-scale burning, but fires can have many origins: natural lightning-induced wildfires, human-set land-clearing fires, or peat and marsh fires that smolder and leave widespread carbon. Distinguishing a single planet-spanning conflagration from regional increases in burning requires stratigraphic breadth, consistent dating, and an explanation for how a regional signal became global. In most examined cases, the burning signatures are patchy, regionally variable, or better explained by more ordinary processes. What does this careful reading yield? The story that emerged in the literature over the past years is not a tidy affirmation of the impact hypothesis. Instead, it is a contested debate where many specialists, sedimentologists, geochemists, archaeologists, have pointed out methodological issues, problems of stratigraphic control, dated materials not directly associated with purported markers, sampling bias towards sites that show an effect, and failures to replicate key tracers in independent cores. Where evidence remains robust, it is often complex and local rather than global and unequivocal. Alternative explanations: increased wildfires driven by drier, windier conditions, local industrial-type processes in much more recent sediments, reworking of older materials into younger layers, can sometimes account for observed anomalies without invoking an extraterrestrial trigger. That does not mean the idea was without value. It prompted useful, rigorous follow-ups, more systematic sampling, improved geochemical protocols, and stricter demands on stratigraphic context. In other words, the claim catalyzed better science. The outcome, as is often the case, is not a dramatic single verdict, but a refining of hypotheses. Some sites show intriguing anomalies that deserve further study, others do not. The net pattern tilts toward caution about asserting a single global impact event as the primary cause of the Younger Dryas. This discussion offers a methodological lesson. Extraordinary, dramatic hypotheses demand extraordinary chains of evidence. Precise dating that ties markers to a single horizon, replicated finds across independent labs and sites, and mechanistic plausibility that links a proposed event to observed climatic shifts. Where one or more of these links is weak, the overall inference weakens. We must also separate the question of an impact from the question of the Younger Dryas's causes. Even if some sites preserve exotic markers, it is a further step to argue that such an event drove global atmospheric and oceanic change. Many plausible climate mechanisms, meltwater pulses altering ocean circulation, internal variability in the coupled ocean-atmosphere system, and volcanic forcing, remain robust candidates because they have mechanistic pathways that climate models can test and that align with multiple independent archives. Finally, an ethical note. Dramatic narratives about comet strikes and sudden disasters can capture public imagination, but they can also mislead when the evidence is preliminary. As listeners, we have a responsibility to prefer careful inference over spectacle. The past is not a movie, it is a set of testable claims built on layers of measurement. The best way to honor human lives in the archaeological record is to be rigorous about what we claim and transparent about what remains uncertain. In the next part, we'll return to the physical archives and compare marine cores and ice cores side by side, their agreements, their differences, and how together they refine our timeline and mechanisms for the Younger Dryas. Dim the lights and imagine two very different libraries. One is a slow white cathedral of ice in the polar north, its pages written in seasonal chemical cycles. The other is a dark sediment-strewn archive on the seafloor, its pages composed of shells, mud, and the detritus of tiny organisms. Both keep time, but they use different alphabets. Tonight we place them side by side and listen for agreement. Start with the strengths. Greenland ice cores give us exceptional temporal precision in the late Pleistocene and early Holocene. Where annual layering is preserved, we can count year by year. The ice records direct samples of ancient atmosphere and trapped gas bubbles, letting us read greenhouse gas concentrations and abrupt shifts in methane and CO2 with remarkable clarity. Those gas records provide global tie points. A methane spike seen in Greenland should also appear in Antarctic ice, with appropriate phasing, because methane mixes relatively quickly through the atmosphere. That property makes methane a particularly useful synchronizing marker across hemispheres. Marine sediment cores offer complementary advantages. They capture ocean temperature changes, salinity signals, iceberg-rafted debris, and the ecological shifts recorded by foraminifera and other microfossils. Some marine proxies, such as sortable silt metrics, P/38 ratios, a chemophysical tracer linked to deep ocean flow, and the composition of benathic foraminifera, speak directly to ocean circulation strength and iceberg activity. Where ice cores tell us the atmosphere cooled in the north, marine cores help show whether and how the ocean redistributed heat and fresh water in ways that could have driven or amplified that cooling. The first practical step in comparison is synchronization. How do you match a layer in an ice core to a layer in a marine core? There are several robust strategies. Tephrochronology can do it when the same volcanic ash appears in both archives. Methane tie points work when abrupt methane shifts appear in ice and can be correlated to terrestrial or marine records that independently track atmospheric methane-sensitive processes. Radiocarbon dating anchors terrestrial and near-shore marine records, though marine radiocarbon requires careful reservoir corrections. Together, these approaches build an inner archive timeline. What do the paired records show for the Younger Dryas? In the North Atlantic realm, Greenland ice documents a sharp V18O decline marking rapid cooling. Many nearby marine cores register contemporaneous drops in sea surface temperatures, changes in foraminiferal assemblages, and increases in ice-rafted debris, evidence for colder seas and enhanced iceberg activity. In some cores, geochemical indicators suggest pulses of freshwater input around the onset, consistent with meltwater routing scenarios. These parallel signals strengthen the interpretation that oceanic changes played a significant role in northern cooling. Yet, caution is required. Marine records are spatially heterogeneous. A core from the Labrador Sea or the Norwegian margin may show strong, immediate responses to northern meltwater pulses. A core from farther south in the mid-Atlantic or from the Mediterranean may record a delayed or smoothed response. Resolution matters too. Many marine records integrate decades to centuries per sample layer because of slower sedimentation rates and bioturbation. That temporal smoothing can mask the sharpness that ice cores reveal at annual resolution. In short, agreement in the broad pattern exists, but precise phasing can differ depending on location and proxy type. The bipolar seesaw concept helps reconcile some of this variability. When the North Atlantic cools because AMOC weakens, less heat is transported north. That tends to cool the northern hemisphere, but can lead to relative warming in the southern hemisphere on multi-decadal scales as heat redistributes. Antarctic ice cores often show a muted or opposite pattern to Greenland on these timescales. Comparing Greenland and Antarctic cores, scientists see interhemispheric seesaw behavior during millennial events. The north plunges while the south changes less or warms slightly. Synchronizing those changes uses gas records, methane, CO2, and volcanic tephras as global markers, showing that the onset of abrupt northern cooling and the southern responses are part of a coupled global system, not a wholly regional anomaly. Proxy-specific challenges complicate direct comparisons. The 18O in ice is primarily a temperature proxy for the source region of precipitation, while foraminiferal D18O integrates temperature and seawater, D18O linked to salinity. Disentangling these signals requires multiple proxies within marine cores. MgCa ratios for temperature, paired D18O for salinity inference, and species turnover data to record ecological change. Pay/Theath ratios hint at how vigorously the deep ocean was ventilated, and thus at AMOC strength, but their interpretation depends on sediment focusing and particle flux. Sortable silt can indicate current strength, but responds to multiple processes, including sediment supply changes produced by meltwater pulses. Dating uncertainty presents another layer. Ice core chronologies, while precise, still require careful corrections for gas age, ice age offsets. The air trapped in bubbles can be younger than the surrounding ice because of diffusive processes in the firn. Marine radiocarbon ages must correct for reservoir effects that vary regionally and through time, introducing potential offsets of decades to centuries. Bayesian chronological modeling is often used to integrate these dates with stratigraphic constraints, but the output depends on the quality and distribution of inputs. When marine and ice archives disagree at the level of a few centuries, interpretation requires humility. Are we seeing true leads or lags, or sampling and dating artifacts? Despite these complexities, an emergent consensus has taken shape. The Younger Dryas was marked by an abrupt northern cooling visible in Greenland, accompanied regionally by oceanic changes that include increased iceberg activity and altered circulation patterns. The timing of onset appears rapid in the north, years to decades, and the termination, the return to warmer conditions, was also abrupt in many records. The precise external trigger remains debated. Meltwater routing, internal ocean atmosphere variability, volcanic forcing, and other ideas each hold explanatory power in some models and archives. The convergence of multiple independent proxies across ice and marine records, however, supports the robustness of the climatic shift itself. Where consensus fades is in the degree to which a single mechanism can explain all observations everywhere. Some cores suggest clear freshwater pulses, timed with onset, others show cooling with less obvious meltwater signals. Regional responses in vegetation, hydrology, and human behavior mirror this heterogeneity. The best current framing respects both the sharpness of the event in northern atmosphere and the spatial complexity of oceanic and terrestrial responses. In the next part, we will dive deeper into isotopic tools, ancient DNA, and tephra fingerprints, how they refine regional narratives and let us trace water sources, population movements, and explosive eruptions with greater confidence. Dim the lights and breathe slow. Tonight we sit with some of the sharper instruments in the scientific toolbox, the ones that let researchers trace water sources, follow human movement, and match a single volcanic eruption across oceans. These methods are highly complementary. Isotopes tell us about origins and pathways. Ancient DNA reveals relationships and migrations, and tephrochronology provides geological barcodes for synchronizing distant archives. Together, they let us move from pattern toward process. Start with isotopes. Isotopes are versions of the same element with different numbers of neutrons. A few isotope systems are especially useful in paleoclimate and archaeology. Strontium, our 87S or 86R, maps geology. Soils and bedrock leach strontium into groundwater and plants. People and animals incorporate that strontium into teeth and bones. Because different bedrock types have distinctive strontium ratios, measuring 87 SAR, 86 SAR in tooth enamel can identify where someone grew up relative to where they were buried. If an adult's teeth and bones show different strontium signatures, that signals movement between geological provinces. In the Younger Dryas context, strontium helps test whether communities relocated, incorporated outsiders, or maintain local continuity. Oxygen isotopes, the 18O, in tooth enamel and speleothems record aspects of water and climate. Precipitation, the 18O, varies with temperature, altitude, and distance from moisture sources. Changes in D18O in speleothems or in tooth enamel can indicate shifts in rainfall patterns or seasonality, useful for tracking whether a locality became drier, colder, or experienced altered moisture sources during the Younger Dryas. Neodymium, Nd, and lead, Pb, isotopes act as tracers of provenance in marine systems. End isotopic signatures in seawater reflect inputs from continental runoff and help fingerprint where freshwater and sediments came from. In marine cores, shifts in Ndod can indicate whether meltwater originated from North America or Eurasia, and Pb isotopes can complement that picture. These systems help test meltwater routing hypotheses by offering geochemical clues about source regions. Carbon and nitrogen isotopes, D13C, D15N, already discussed for diet, also inform ecosystem baselines. They reveal whether primary production leaned more on C3 or C4 plants, and whether food webs shifted in trophic structure. When paired with other proxies, they let us infer broader ecological reorganizations. Isotope systems are powerful, but interpretation requires caution. Multiple processes can produce similar isotopic signals. Mobility averages over tissue turnover times, tooth enamel records, childhood, bone collagen, a later life average, and diagenesis can alter original ratios. Good practice pairs isotopic data with archaeological context, other chemical tracers, and clear sampling protocols. Now, ancient DNA, aDNA. Over the last 2 decades, aDNA revolutionized how we read past human population dynamics. Genome-wide data can reveal ancestry, kinship within burials, population continuity or replacement, and sometimes even pathogen DNA preserved in teeth or bone. For the Younger Dryas interval, aDNA studies have clarified regional patterns. In some areas, local genetic continuity persists across the climatic shift. In others, signals of admixture or movement appear. That complexity mirrors the archaeological mosaic of continuity and change. Beyond ancestry, aDNA can identify small-scale demographic events. Reductions in genetic diversity may indicate population bottlenecks. Kinship within burial clusters can reveal household or lineage organization, and pathogen DNA, when preserved, can reveal disease presence that might intersect with environmental stress. But aDNA comes with caveats. Preservation bias, cold, dry, or sheltered contexts preserve DNA better. Sampling bias, sites and individuals sampled are not random. And the ethical imperative to treat human remains with respect and often in consultation with descendant communities. Proteomics and ancient proteins offer complementary reach where DNA fails. Collagen peptides and other proteins survive in contexts too warm for good DNA preservation. Protein evidence in dental calculus or on residues can confirm species-level identifications. Milk proteins point to dairying, cereal storage proteins indicate specific grains, and so on. These molecular identifications strengthen behavioral inferences about diet and subsistence. Tephrochronology is the geologist's barcode. A volcanic eruption ejects glass shards and mineral crystals whose chemistry is diagnostic of that eruption. When tephra falls into lakes, onto ice sheets, or into soils, it forms a thin but widely dispersed layer. If the same tephra appears in an ice core, a marine core, and an archaeological sequence, and its geochemistry is matched carefully, it provides a synchronous tie point. Tephra matching relies on precise geochemical fingerprinting, major and trace element analysis of glass and minerals, and on robust stratigraphic context. Tephra is especially valuable when an eruption can be independently dated or when it appears in multiple archives that otherwise have different dating systems. Combining these methods creates robust narratives. For example, a speleothem dated by Yupuyu Theish that shows a sudden shift in D18O can be tied to an ice core methane event via tephra or gas matching. Strontium and Ned-D isotopes can show whether runoff sources changed at the same time. ADNA from nearby human remains can reveal whether population structure shifted. That multi-proxy synchrony is the gold standard, independent methods converging on a common timeline and mechanism. Limitations remain. Each proxy carries its own timescale and resolution. Teeth-lock childhood, speleothems may average seasonal signals, marine sediments can integrate decades, reservoir effects, diagenesis, and post-depositional mixing complicate straightforward reading. Statistical frameworks, especially Bayesian models, help integrate diverse dates and their uncertainties, but they cannot substitute for careful, stratigraphic control and thoughtful sampling design. An ethical note is essential. Many of these techniques require destructive sampling, drilling teeth, shaving speleothems, or taking sediment slices. Modern practice emphasizes minimal sampling, transparent justification, and consultation with local stakeholders and heritage authorities. Respecting cultural contexts and ensuring that scientific gain does not come at the expense of people's heritage is now standard in responsible research. Finally, consider what these methods reveal together for the Younger Dryas. Isotopes help identify shifts in water sources and possible meltwater pulses. ADNA maps population continuity and movement. Tephra synchronizes distant records. Proteomics and residue work confirm dietary choices. When these lines align, the case for a particular mechanism or social response strengthens. When they diverge, the discrepancy points to complexity and prompts new hypotheses. In the next part, we'll take this multi-tool perspective and apply it regionally. The Fertile Crescent, Europe, and the Americas. How each landscape and its people experienced and responded to the Younger Dryas in distinct but interconnected ways. Dim the lights and let your attention travel like a slow hand across a map. We will move region by region, listening to the particular voices each landscape keeps. The Younger Dryas is a global whisper with local dialects. In some places it sounds like a hard winter, in others like shifting coastlines, and in others like changes in who is living where. We'll keep the cadence slow, image, evidence, caution, and pay attention to what the archives actually say. The Fertile Crescent, river margins, terraces, and grain patches begin in the places most central to our question about agriculture, the Levant and the Fertile Crescent. Here, semi-sedentary communities already practiced concentrated gathering, storage, and localized cultivation of wild cereals. Archaeology at sites in this region records durable houses, storage pits, grinding suites, and charred cereal remains that become more prominent through the interval that brackets the Younger Dryas, roughly 12 to 109 and 1100 years before present. What changes do we see? Palynology in nearby lake cores and off-site sediments often records a shift toward more open, steppe-like vegetation and a reduction in woodland taxa, signs of cooler, often drier conditions. Locally, however, wetland patches and river margins frequently persist as micro-refugia where wild cereals and wetland resources concentrate. Those pockets matter. People who already tended to harvest along terraces and streams could intensify collection in these refugia. Archaeobotanical assemblages from several Levantine sites show increases in wild cereal macro remains and more grinding equipment, suggesting intensified processing. Isotopic evidence from human remains in some sequences aligns with modest shifts toward greater plant contribution in diets. ADNA from human burials in the broader Near East tends to show regional continuity across the Younger Dryas in many sampled localities, indicating that where people stayed, populations were not completely replaced. Instead, social networks likely reconfigured, more exchange, cautious movement, or local folding together of groups. Tephra correlations and youth-dated speleothems help anchor these local chronologies to the wider climate sequence, strengthening the inference that people in the Fertile Crescent were adapting in the face of real ecological change, not static weather noise. Europe, retreat, refugia, and social reconfiguration. Move northwest to Europe and picture a landscape where forests withdraw from higher latitudes and steppe expands, at least temporarily, in many regions. Pollen records show declines in some temperate trees and increases in herbs and grasses in parts of Central and Northern Europe. Marine cores off the north and northwest coasts record colder sea surface conditions and more iceberg influence than before. Human responses vary by region and by economy. In parts of Western Europe, hunter-gatherer groups show shifts in settlement location and in the spectrum of prey exploited. In some archaeological sequences, there is intensification of foraging breadth, more use of small mammals, fish, and plant foods, while in other places mobility patterns change, with greater seasonal aggregation or dispersal, depending on local resource reliability. ADNA studies across Europe reveal a patchwork. Some regions show continuity of lineages, others episodic influxes or gene flow consistent with population movements, but the picture is complex and localized rather than uniform. One major and sensitive topic in North American and European studies is megafaunal extinction timing and causation. While some megafauna disappearals cluster around the terminal Pleistocene, the role of climate versus human hunting versus other stresses remains debated and regionally variable. For Europe, changes in prey communities and human strategies often reflect a web of ecological and social responses more than a single cause. The Americas, meltwater routes, Clovis, and varied impact across the Atlantic, North America presents its own patchwork. Some eastern and mid-continent lake cores preserve black mat layers and changes in pollen and charcoal that have attracted intense attention. In the plains and east, archaeological change around the Younger Dryas interval is complex. Clovis and other early Paleo-Indian technologies show transformations, and megafaunal declines in some regions temporarily overlap with the climatic window. Claims tying a single cosmic impact to widespread extinctions and cultural change have been scrutinized, as we discussed earlier, and the mainstream view emphasizes multi-causal explanations. Climate stress, human predation pressures, habitat change, and regional ecological dynamics interacting in space and time. Proglacial lakes in North America, especially Lake Agassiz, are central in meltwater hypotheses. Sediment and geochemical proxies from marine cores and continental outflow deposits sometimes record large discharges that could route fresh water into the North Atlantic via river systems. Where marine proxies show freshwater pulses, timing often aligns broadly with Younger Dryas onset, supporting the idea that meltwater routing contributed to ocean circulation change. But the marine picture is spatially heterogeneous, and not all ocean basins or cores record the same clear pulse magnitude. Regional synthesis. Heterogeneity is the rule. A steady theme emerges in these snapshots. The Younger Dryas is not a uniform global winter. It is a rapid and regionally variable climate event that alters water budgets, vegetation, and animal communities in ways that depend on local geography and ecology. The human response is equally variable. In the Fertile Crescent, pre-existing investments in storage and plant processing offered pathways to intensify cereal use and persist in place. In parts of Europe and North America, greater mobility, shifts in prey spectrum, and local relocation were common strategies. These are not alternative narratives of failure or success. They are contingent, pragmatic responses shaped by culture, prior investments, and the immediate resource map. Evidence integration. What strengthens region-level claims? We rely on converging lines of evidence to build regional stories. Pollen and macrobotanical remains for vegetation and plant use, marine cores and isotopes for ocean forcing, speleothems and lake varbs for local hydroclimate, archaeological features for subsistence and settlement, ADNA for population continuity or movement, and tephra for synchronization. Where multiple archives align in timing and direction, our confidence grows. Where they diverge, we must be cautious and explore why. Sampling, dating offsets, or truly different local responses may be at play. Uncertainties and sampling bias. Remember two critical cautions. First, sampling bias shapes our view. Well-studied regions with preserved organic archives dominate narratives. Many landscapes leave poor records. Second, dating uncertainties at the decade to century scale matter when we ask whether human changes were synchronous with abrupt climate steps. Bayesian modeling helps, but it depends on quality inputs. So, regional snapshots are provisional, robust in general pattern, but fluid in detail. Sit with that variability a moment. People in different places felt the Younger Dryas differently. For some, it was an added burden on long-tested routines. For others, an accelerator of change. In the next part, we will turn specifically to agricultural beginnings, how people intensified plant use, the archaeological signatures of early cultivation, and why the timing and meaning of those signs matter for our broader question. Dim the lights. Let your breathing slow, and imagine a small, warm room where flower dust hangs in the air like a faint, pale mist. A stone sits steady in a hand. The rhythmic scrape of a mill repeats, steady as a metronome. These are the quiet, repetitive technologies that first made seed into daily bread, and in doing so, rewired human timeframes. When we speak of agricultural beginnings, we do not mean a single moment when people flipped a switch and became farmers. Rather, think of a long conversation between people and plants in which small, repeated choices, where to harvest, which plants to favor, how much to store, gradually accumulated into new practices. These practices leave signatures in the ground. Our job as listeners and interpreters is to read those signatures carefully and separate preparatory behaviors from full domestication. The first line of evidence is the toolkit of intensification. When you find a higher density of grinding stones, paired with more and better sickle implements, you are seeing labor invested into plant processing at a scale beyond opportunistic gathering. Sickle blades with characteristic silica polish, sickle gloss, show repeated cutting of silica-rich grasses. Wear patterns on hafts and residues on blade surfaces preserve microfossils that directly indicate serial processing. When grinding stones proliferate within a house floor or courtyard, they tell of routine, community-scale processing rather than occasional use. Next, plant remains themselves. Charred seeds are the most direct and stubborn witnesses. Burning preserves organic material well. Hearth contexts thus act as accidental museums. Two morphological changes particularly matter for archaeobotanists as signs of domestication. The first is seed size. Many domesticated cereals show directional increases in seed dimensions over time compared with their wild ancestors. The second is the rachis structure, how the seed head detaches. Wild wheats and barleys have brittle rachises that shatter easily to disperse seed. Early domesticates evolved tougher rachises that retain seeds on the ear, a trait attractive for harvesting and threshing. Tracking these traits in stratified samples provides a temporal arc from wild collection toward managed sowing and selection. But morphology is only part of the story. Phytoliths, microscopic silica bodies from plant tissues, are durable and can record the presence of grasses, cereals, and even specific plant parts, stems, husks, leaves. Phytolith assemblages and occupation layers, on tools, and in dental calculus help confirm the presence and processing of cereals even where charred macro remains are rare. Starch grains trapped in grinding stones and dental calculus are another direct line of evidence. They identify which plants were processed and eaten. When multiple molecular lines, charred seeds, phytoliths, starches, and protein residues point to the same taxa, the inference is strong. Experimental archaeology gives texture to those signals. When researchers grow wild cereals under different management regimes, harvesting by hand, minimal or no tillage, deliberate sowing in cleared patches, they observe how plant populations respond. Seed size may shift, weed suites change, and yield patterns vary. These experiments show that even low-intensity human tending, repeatedly harvesting and favoring seed from the largest heads, can produce measurable evolutionary shifts across only a few human generations. In short, selection need not be conscious or systematic to change plant traits. Repeated small-scale human behaviors can do the work over centuries. Landscape context matters too. Terracing, simple soil preparation, and the creation of disturbance patches near dwellings, along irrigation channels, or on river terraces, create favorable niches for wild cereals that prefer disturbed soils. Archaeologists identify such landscape modifications in the form of field terraces, clearance features, or concentrations of burned patches. Paleoclimatological data from nearby sediments can show increases in cereal-type pollen, though pollen's transport makes local inference trickier than direct macro remains. Social and economic signatures are equally instructive. Storage pits and granaries indicate not only surplus, but the social frameworks to protect and distribute it. Storage changes social temporality. The household that can save food across seasons becomes able to plan beyond immediate scarcity. At the same time, surplus invites new social arrangements, inequalities of access, obligations of redistribution, and new trade relationships. Archaeological features, specialized storage architecture, more substantial hearths for processing, and designated communal workspaces hint at these social shifts. We must also watch genetic signals. Ancient DNA from plant remains is increasingly available and provides powerful evidence for selection. Genetic markers of domestication, mutations associated with non-shattering or larger seed size, can sometimes be identified and dated. When genetic change aligns with morphological and contextual evidence, more processing tools, storage structures, the case for managed cultivation strengthens. Proteomics, detecting seed storage proteins, can confirm which cereals were processed, and sometimes whether they were consumed as stored foods or fresh harvests. But the record is nuanced and uneven. In many places we see a prolonged pre-domestication cultivation phase, people tending, harvesting, and processing plants intensively without immediate widespread morphological domestication. In other words, behavioral intensification can predate clear morphological domestication by centuries. The distinction matters because it reframes the dawn of agriculture as a long, regionally variable process of human-plant interaction rather than a single technological revolution. Uncertainty boxes that matter here. 1. Chronology. How tightly can we align increases in plant processing with abrupt climate markers? Radiocarbon uncertainties of decades to centuries make exact phasing difficult. 2. Equifinality. Similar archaeological signatures, e.g., increased cereals, can arise from different causes. Deliberate cultivation, opportunistic intensification, or environmental shifts that temporarily concentrate wild stands. 3. Preservation bias. Charred seed, survival is patchy. Organic artifacts are often absent in many sites, obscuring behavior. 4. Sampling bias. Well-studied core areas, like parts of the Levant, may overrepresent regional trajectories. Ethical note: plant remains and soils are not just data. They often come from landscapes still important to living descendant communities. Excavation and destructive sampling require consultation, minimal intervention, and transparent data sharing. Many projects now embed local stakeholders in research design to ensure respectful handling of landscapes and finds. For you, listening now, picture the simple, repeated acts that accumulate into agriculture. A family planting a handful of seed where last year's store was abundant. A child carrying a heavier basket to a communal mill. A household carefully sealing a pit against damp. These small, ordinary choices, repeated and social, are the ingredients of deep transformation. In the next part, we will step back and honestly list the open uncertainties, the precise timing questions, the causal threads we can't yet untangle, and the types of new data that would clarify them. Dim the lights and let your breathing be steady and unhurried. Sit with the feeling of a slow clock whose hands sometimes blur. Ancient records whisper about events that happened long ago, but their voices are not always in perfect agreement. In this part, we'll hold those disagreements openly and gently, not to undermine what we know, but to be clear about the edges where certainty thins and curiosity should deepen. One of the first and most persistent uncertainties is timing, at the level that matters most to human lives. The difference between an event happening within a generation and it unfolding over many generations. Greenland ice cores can speak year by year in places. Some lake varves can do the same. Many radiocarbon-dated archaeological sequences, however, come with uncertainties that span decades, or even a century. That gap is meaningful. If a cooling pulse arrives within a decade, a household might make different decisions than if the same cooling unfolds over a century. Right now, aligning narrow, high-resolution archives with the archaeological record still challenges us. We can often say that climatic cooling and changes in plant use broadly overlap in time, but in many local cases we cannot yet claim decisive, year-for-year causality. The result is a landscape of plausible interactions, rather than a single, neatly timed cause and effect narrative. Closely related is the question of spatial heterogeneity. How uniform was the Younger Dryas across the Fertile Crescent or across Eurasia? The pollen and lake records show coherent trends in many places, but they also show local refugia where conditions differed enough to sustain wetland plants or cereal stands. For human communities, those micro-refugia could be the difference between staying and moving. Yet our sampling is uneven. Lakes, caves, and well-preserved middens are not distributed evenly across the land. And preservation biases mean that some of the most informative microenvironments are underrepresented. Until we fill geographic gaps with comparably high-resolution records, our regional stories must stay candid about where inferences are robust and where they remain tentative. There is also a methodological knot around marine reservoir effects and reservoir age variability. Surface ocean radiocarbon ages change with circulation patterns, upwelling, and freshwater input. When those ocean conditions themselves are what we are trying to test, the corrections become circular and uncertain. This affects not only pure marine records, but also nearshore and estuarine samples that might feed into archaeological chronologies. Improving regional reservoir corrections and developing independent, non-radiocarbon tie points, such as tephra layers, VARV counts synchronizable to ice core gas markers, and more euthid-dated speleothems, would narrow this knot, but those data are not yet abundant in every key region. The mechanism question remains another honest place of ambiguity. Melt-to-water routing that alters Atlantic circulation is a plausible and model-supported pathway to northern cooling. Yet other mechanisms, large volcanic eruptions, changes in sea ice extent amplifying regional cooling or internal ocean atmosphere variability, also have traction, and different archives emphasize different parts of the story. The debate is not merely academic. Distinguishing between a sudden, externally forced pulse and an internal threshold crossing changes how we conceive of impact on hydrology, on vegetation response rates, and on human agency. Right now, models and proxy records often point to a multi-causal picture. Meltwater pulses likely mattered in some regions and times, and they may have been sufficient to push a system already primed toward abrupt change. But proving the priming, the tipping, and the relative weight of each forcing in every place is still work in progress. A fourth uncertainty is behavioral. How uniform were human responses, and to what extent were changes archaeologically visible? We see increased grinding stones and storage in many Levantine sites, but those signatures can mean different things in different contexts. A strategy of planned storage to ride out surprising bad years, a community-level adaptation to a longer-term risk, or a social display of prestige linked to accumulation. Similarly, mobility signatures in isotopes and settlement patterns can signal forced movement, seasonal decisions, marital practices, or long-term reorganization. Parsing social meaning from material patterns requires careful, context-sensitive interpretation, and often a richer set of complementary data, including household-level sampling, more ADNA that reveals kinship patterns within burial clusters, and ethnographically informed analogs that can guide plausible readings without imposing modern narratives. There are also laboratory and sampling uncertainties that quietly shape interpretations. Contamination in radiocarbon samples, open system behavior in euthyath samples, or diagenetic alteration in proteins and DNA can produce misleading ages or false negatives. Some dramatic claims in the past have faltered because independent labs could not reproduce exotic markers. The remedy here is methodological humility. Replication, standardization of sampling and pre-treatment protocols, blind testing where possible, and the routine archiving of samples so others can re-examine them with improved techniques. A related ethical uncertainty deserves explicit mention. Many of the methods that would reduce uncertainty require destructive sampling, micro-drilling speleothems for UT dates, taking tiny chunks of well-preserved bone for ADNA, sampling sediment columns from cherished landscapes. The ethical calculus involves balancing the scientific value of data against the duty to protect cultural heritage and respect descendant communities. The research community has made progress, minimizing sample sizes, transparent reporting, and collaborative project design, but the tension remains. A richer, ethically informed research agenda that honors local stakeholder voices will both strengthen science and guard against harm. So, what kinds of new data would most help resolve these uncertainties? Higher resolution, well-dated lake cores from undersampled parts of the Fertile Crescent. More the dated speleothems that directly record local hydroclimate with precise ages. Expanded networks of VARBed sequences that can be counted year by year and tied to ice core gas markers. More cross-checked tephra correlations. And dense, regionally distributed aDNA sampling done with ethical frameworks in place. Improved regional reservoir corrections and concerted replication studies of contentious proxies would also be transformative. Equally important are comparative multidisciplinary field programs that design sampling strategies to answer the timing and causation questions explicitly, rather than assembling proxies opportunistically. Finally, hold a thought about uncertainty as a kind of invitation rather than a failure. The places where the map is pale are the places where curiosity can do meaningful work. Scientists do not treat uncertainty as a reason to stop. They treat it as a reason to collect better, to replicate, and to refine both methods and narratives. For listeners, that stance is a modest encouragement. The past remains knowable in ever finer resolution as colleagues bring better data sets and better ethics to the task. In the next part, we will continue this theme of open questions by turning to modeling and competing explanations. What climate models reveal about thresholds, and how they test the plausibility of meltwater pulses, volcanic forcing, and internal variability. Dim the lights and let your breath find its slow, even rhythm. In the last part, we held the open questions in our hands. Now we put them into the workshop of climate models to see how theory and simulation press against stubborn data. Models are the thought experiments of climate science, simplified worlds in which we can tweak fresh water, add volcanic aerosols, or block parts of the ocean and watch how the system responds. They do not prove history. They test plausibility. Where models and proxies agree, confidence grows. Where they diverge, the tension points to missing pieces in either the simulation or the record. Start with the simplest modeling intuition. The Atlantic overturning circulation, AMOCs, relies on dense surface waters forming in the high latitudes. That density depends on both temperature and salinity. Add a lot of fresh water to the surface, and it resists sinking. In many models, a sufficiently large, rapid freshwater pulse to the North Atlantic reduces deep water formation and weakens AMOC. The atmosphere above cools. Sea ice expands. These changes feed back into the system, changing winds, precipitation, and even subtropical rainfall belts. That chain of cause and effect is direct and mechanistic. Freshwater input affecting ocean density changing heat transport, and altered heat transport changing regional climates. But the devil is in the details. Models disagree about how much fresh water is required, where it has to enter, and how the system was configured before the pulse. Some models show dramatic AMOC weakening, with modest freshwater forcing when the initial state is already near a threshold. In other words, the system is primed to tip. Other models require much larger pulses to produce comparable effects. Differences stem from model resolution, the complexity of ocean ice atmosphere coupling, parameter choices for mixing and convection, and how sea ice processes are represented. Some models include dynamic ice sheet components and realistic iceberg trajectories. Others treat freshwater as a simple surface flux. These structural choices matter: how an iceberg melts, where its freshwater disperses, and how quickly it mixes into the upper ocean, all change the model outcome. There is a second family of modeled explanations. Volcanic forcing can inject aerosols and ash into the atmosphere, reducing solar radiation at the surface for seasons to years. Several modeling experiments show that clusters of large eruptions, properly timed and located, can produce short-term cooling that might nudge ocean ice systems into altered states. Yet volcanic scenarios struggle with persistence. Aerosols fade, but the Younger Dryas persisted for roughly a millennium. For long-duration cooling, models generally need sustained forcing or internal feedbacks that maintain the cold state after the initial push. That is why volcanic-only stories have limited traction, unless coupled to other mechanisms or invoked as part of a sequence. A third set of models explores internal variability, modes of the coupled ocean-atmosphere system that can shift without an external shove. In these simulations, the system can reorganize because of non-linear internal dynamics. If the climate was already cooling at the end of the last glacial internal oscillations, could amplify the trend into a prolonged cold phase. Models of this type highlight that not every abrupt event requires an obvious external driver. Complex systems can produce abruptness from within. The challenge here is deciding whether internal thresholds were realistically accessible under the boundary conditions of the late Pleistocene. A related issue is the spatial routing of fresh water. If Lake Agassiz drained into the Arctic via the Hudson Strait, the fresh water would affect different parts of the North Atlantic than if it drained through the Mississippi and Gulf of Mexico. Models that explicitly route fresh water down particular channels produce different regional fingerprints. That variability matters when we try to match model outputs to Marine Corps signals. A model that puts fresh water into the Labrador Sea may compare well to certain cores and poorly to others. Thus, agreement with one set of proxies can coexist with mismatch to another, simply because of routing decisions. Model disagreement also arises from how they treat sea ice. Sea ice is not passive. It changes albedo, modifies heat exchange, and alters ocean atmosphere momentum. Some models represent sea ice with simplified thermodynamics and brittle mechanics. Others simulate dynamic drifting flows with melt ponds and leads. Those differences influence how quickly sea ice expands during a freshwater-induced cooling and how persistent that expanded cover remains. If sea ice expansion is large and persistent, it reinforces cooling and can prolong the cold phase beyond the freshwater pulse. Parameter sensitivity is another key source of difference. Small changes in mixing coefficients, convection thresholds, or cloud feedback strengths can yield qualitatively different outcomes. Model ensembles, running the same model many times while varying parameters within plausible ranges, reveal the range of possible behaviors. In many model intercomparison projects, ensembles show both that AMOC collapse is plausible under certain forcings and that a non-collapse, milder cooling is also possible. The ensemble approach is therefore instructive. It replaces single model certainty with a probabilistic map of plausibility. So, where does that leave us relative to the Younger Dryas? Models collectively suggest that meltwater forcing is a credible and mechanistically plausible pathway to abrupt northern cooling, and that such a pathway is reinforced if the system was already primed by other factors. Models also show that volcanic or internal variability alone can, in some simulations, produce significant cooling, but face challenges explaining the duration and hemispheric pattern. The most defensible stance at present is multi-causality. Several forcings and state-dependent internal dynamics likely interacted to produce the observed pattern. Models do not settle this with a single bullet. They map plausible routes and identify the types and amounts of forcing each route requires. What would sharpen the modeling inferences? Two improvements stand out. First, better boundary conditions anchored to data. Higher resolution reconstructions of ice sheet geometry, freshwater volumes, and meltwater routing would reduce guesswork. Second, more and better temporally constrained proxy data that can be directly compared to model outputs at matching timescales. For example, high-resolution marine cores that record local salinity and temperature, tephra-synchronized across basins, would let modelers test whether a simulated freshwater pulse produces the same regional pattern of change. Model data assimilation frameworks that iteratively nudge models toward proxy-informed states are promising here. We must also acknowledge a philosophical limitation. Models simplify. They necessarily omit some processes or represent them at coarse scales. That is not failure. It is the nature of modeling. The question is not whether models are imperfect, but whether they are useful. In the Younger Dryas case, models remain indispensable. They show what is physically plausible, what combinations of forcings could produce observed patterns, and which pieces of proxy evidence most constrain the possibilities. Listen to that. Models as maps, not verdicts. They help narrow where to look and what to measure next. In the next part, we will move from the scientific workshop back to human stories, how societies reorganized, who moved, and what archaeological patterns tell us about social memory and exchange in the Younger Dryas world. Dim the lights, breathe slowly, and imagine a slow braid of footsteps stretching across a landscape. A family-led walk to a seasonal pasture, a group carrying stored grain to a neighboring household, traders moving obsidian along long-established routes. Societies are not fixed pictures. They are processes of motion, memory, and exchange. In this part, we follow those movements, of people, of objects, of stories, and listen to the traces they left in stone, bone, and glass. Start with mobility in its many registers. Movement can be seasonal and planned. Flocks moved to higher pastures in summer and back in autumn. Households that followed fish runs or plant ripening on a known cycle. It can be opportunistic, people relocating after a harvest failure, or groups merging when resources concentrated in refugia. Mobility can also be episodic and forced. Conflicts, disease, or sudden environmental declines that push people to seek new lands. Archaeologists read mobility through several signals: shifts in raw material sourcing, isotopic differences between childhood and adult tissues, changed settlement footprints, and modified toolkits that reflect new resource focuses. Obsidian sourcing is a clear material indicator of long-distance ties. Obsidian, volcanic glass, has chemical fingerprints tied to specific outcrops. When a blade at a Levantine site matches an Anatolian source, it speaks of networks that linked people across hundreds of kilometers. During times of stress, these networks can tighten, more frequent exchanges to secure scarce goods, or they can fray, leading to more local procurement and reduced long-distance trade. The archaeological record shows both patterns regionally. Where exchange continued, communities could buffer shortages. Where it collapsed, reliance on local resources intensified. Isotopic analyses of human teeth and bones give a personal scale to movement. Tooth enamel preserves a childhood signature. Bone collagen averages the last decade or two of life. When those signatures differ, a life story emerges, a person born in one geological catchment who later lived and died in another. In several Younger Dryas contexts, isotopic work reveals both continuity, many individuals showing local signatures, and movement, some individuals clearly non-local. These patterns suggest flexible social strategies, intermarriage with distant groups, seasonal migration of certain households, or the incorporation of outsiders during difficult times. ADNA adds another dimension. Where genetic sampling is dense enough, researchers detect patterns of continuity and admixture. In places of long-term occupation, local genetic continuity often persists despite environmental stress. In other locales, signals of admixture or new ancestries appear, suggesting movements of people. Combining ADNA with isotopes and burial context is especially revealing. Kin groups buried together, combined with isotopic diversity, can show both household cohesion and wider social connections. But remember the caveats. Preservation and sampling bias can skew our view. ADNA snapshots are powerful but partial. Exchange is also cultural and mnemonic. Objects carry stories across space. Beads, carved bone, specific decorative styles. When a decorative motif appears widely across sites during a difficult period, it could mark shared identity, refugee circulation, or deliberate alliance building. Ritual items and shared burial practices often act as social glue. In houses where secondary burials or ancestral caches appear, we glimpse how memory and place anchored social identity even when mobility increased. People could move but still carry ancestors' names, stories, or objects that signaled belonging. Markets, informal and formal, transform movement into social economics. Small-scale exchange, raw materials for processed foods, crafted goods for labor, can be a stabilizing force. Ethnographically, we see that during lean periods, people often widen reciprocal ties, more frequent, smaller exchanges that create webs of mutual support. Archaeological proxies for such activity include concentrations of non-local items in domestic contexts, the persistence of standardized tool forms that ease trade, and shared manufacturing evidence suggesting specialized producers supplying broader areas. Migration also reshapes social structure. A settling of newcomers into a place with established households can reconfigure land use, craft specialization, and marriage networks. Sometimes newcomers bring useful practices, new stone knapping styles, food processing methods, or social institutions that are adopted locally. Other times, incorporation is partial and fraught, producing hybrid assemblages that mix styles and technologies. These hybrid zones are rich sources of cultural innovation, but can also be sites of tension. competition for resources, renegotiation of access to stored food, and debates over ritual precedents. The archaeology of storage and property rights plays into mobility too. Durable storage, substantial bins, clay-lined pits, sealed granaries encourages stationary investment. Communities with robust storage can plan for risks and may be less likely to uproot at the first sign of scarcity. Conversely, the absence or loss of secure storage can force households into more frequent movement. In the Younger Dryas landscape, we therefore see a mosaic. Some settlements with strengthened storage and heavier processing equipment that suggest a choice to stay and manage scarcity, and other areas showing abandonment or reduced occupation intensity where mobility was the adaptive strategy. Memory and narrative are another vector of movement. Stories travel with people, techniques for bread making, seed selection rules, ritual formulas, and collective memories of past scarcity. These non-material traces leave subtle archaeological impressions. Continuity in pottery styles that suggests cultural transmission, repeated house plans that imply shared knowledge of construction, and persistent burial customs that link generations. Memory can be conservative, anchoring groups to place. It can also be mobile, carried to new valleys by newcomers who recreate familiar social forms. Conflict and cooperation are twin possibilities. Stressed resources can heighten competition, leading to rating or territorial adjustments that push people to relocate. Alternatively, stress can intensify cooperation, alliances for shared water maintenance, combined foraging forays, and marriage ties that knit groups together. Archaeological signals of conflict, mass graves, palisaded sites, trauma on skeletons, are relatively rare in many Younger Dryas sequences, suggesting that large-scale violent breakdown was not a universal outcome. More commonly, the record speaks of small-scale adjustments, negotiated sharing, and the reweaving of social ties. Finally, an ethical reminder. Studying movement touches on ancestry and living descendant communities. Interpretations about migration and exchange can have modern political weight. Responsible research practices include transparent reporting, community consultation, and sensitivity to how narratives about population movement are framed and used. Listen to that braided thread of footsteps again. Movement is not a single pattern, but a spectrum of strategies. Seasonal rounds, long-distance exchange, targeted relocation, and cultural transmission. In the next part, we'll examine ecology and food webs, how cold changed the availability of plants and animals, how trophic relationships shifted, and what that meant for daily caloric economies and material supplies. Dim the lights and let your breath slow. Imagine the landscape as a kitchen table laid out with different bowls. Grasses in one, reeds and tubers in another, herds of ungulates grazing beyond, and streams threaded with fish. When climate shifts, the contents of those bowls change. The size of portions, the regularity of refills, the ease with which hands can reach for them. Tonight we follow those shifting menus, from plants to predators, and listen to how ecosystems recompose themselves when the world cools. A shorter growing season is the most immediate ecological blunt instrument. Cooler temperatures compress the window in which plants can photosynthesize, flower, and set seed. For annual wild cereals, this can mean fewer seeds per stalk, delayed ripening, or a shift in the places where they successfully complete a life cycle. On slopes and terraces, species that once matured reliably may retreat downslope or into sheltered microhabitats. Trees that depend on sustained warm seasons fail to recruit saplings, and woodlands thin. That opens space for steppe and herbaceous communities, grasses, wormwood, and saltbush. Changes that we read in pollen and in the plant remains recovered from archaeological contexts. Wetlands are especially vulnerable. Many marshes, reed beds, and floodplain patches rely on a narrow balance of precipitation, groundwater discharge, and seasonal flooding. Cooler conditions that reduce overall precipitation or alter snowmelt timing can shrink these wetlands, turning once rich patches of tuber-producing and rush-bearing ground into drier, less productive flats. For people, wetlands are concentrated resource hubs. They yield fish, waterfowl, edible roots, building materials for baskets and mats, and attract game. Losing them reallocates calories across the landscape. What was once predictable becomes patchy and risky. Faunal communities respond in linked ways. Large grazing herbivores often depend on a certain biomass of grasses and browse. When plant productivity falls, herd sizes shrink or move to more favorable ranges. Species composition can change. Animals adapted to open, colder steppe may expand, or conversely, species that require particular vegetative cover may decline. Smaller mammals and invertebrates often show more rapid population responses, sometimes increasing where shrubs and grasses create new niches. Predator populations follow their prey. Wolves, large cats, and raptors either decline, shift ranges, or concentrate around remaining refugia, increasing local competition and altering risk landscapes for humans. These shifts create trophic cascades with practical consequences. Less abundant large game increases the search and handling time for high-calorie returns. Hunting strategies may shift from targeting large-bodied ungulates to focusing more on small mammals, birds, fish, and intensified plant processing. That is a change not only in what people eat, but in the economics of foraging. Return rates, calories per hour invested, change, and so do the labor and toolkits optimized for procurement. Grinding stones and sickles become more valuable, where plant returns can be reliably secured. Nets, traps, and fish hooks gain utility where aquatic resources remain accessible. Aquatic systems respond in their own rhythms. Cooler, windier conditions can change stratification in lakes and the timing of plankton blooms, affecting fish productivity. Lower water temperatures sometimes boost oxygenation and benefit cold-adapted species. Other times, reduced nutrient inflow from droughted catchments lowers primary productivity and collapses fish yields. River flows change with altered snowmelt, shifting the seasonality of spawning runs and the availability of fish at traditional harvesting spots. In coastal contexts, altered sea surface temperatures and circulation affect upwelling and plankton blooms, which cascade up to shellfish and fish stocks that coastal communities rely on. The net result for human caloric economies is an increase in uncertainty and a rise in the value of buffering strategies. Buffering can be biological, favoring foods that store well, or social, widening exchange networks and sharing risk across households. Stored grain becomes a kind of temporal currency that smooths seasonal shortfalls. Diversifying the diet reduces vulnerability. A mix of dried fish, stored seeds, preserved meat, and gathered tubers is less likely to fail entirely than dependence on a single resource. That is why archaeological signatures of storage and processing equipment often cluster in places where environmental variability rose. They are adaptive responses to shifting food webs. Yet there is no single human response. In some places, communities intensify plant use because wild stands remain accessible and because processing yields stable returns. In other places, people double down on hunting mobility, following migrating herds. In still others, fish and wetland resources become central. This ecological mosaic mirrors earlier parts of our story. Local refugia matter. A narrow floodplain that keeps its water becomes a lifeline. A patch of south-facing terrace that still warms early may sustain cereals where surrounding slopes cannot. Microtopography and hydrology reshape viability at the household scale. Ecosystems also reorganize in ways that influence non-food material supplies. Shrinking reed beds reduce the availability of thatching and mat materials, affecting shelter and storage technology. Loss of dense wood may change fuel economies, prompting the use of dung, peat, or other combustibles. Raw material flows for toolmaking, flint sources, bone for points, large timber for posts may become harder to access, shifting craft specialization and trade. In short, ecological change rethreads the fabric of daily life, not only what people eat, but how they build, transport, and store. Long-term resilience often depends on social protocols that mediate resource stress. Rules for sharing, taboos that regulate harvest timing, and ceremonies that maintain cooperative labor. Ethnographic analogs show that many societies develop norms: who gets priority access to stored grain, how meat from a large kill is distributed, that reduce the risk of immediate starvation and preserve social cohesion. Archaeology sometimes hints at such protocols through standardized storage features, communal structures, and repeated ritual deposits that suggest shared obligations. These social adaptations are part of the ecological story. A short ethical note is warranted. When we study past ecologies, we often do so in landscapes that remain culturally significant to living communities. Discussions of changing wetland extent, lost pasture, or shifting resource zones intersect with contemporary livelihoods and heritage. Research and storytelling should therefore proceed with sensitivity, recognizing that ecological narratives about the past can resonate in living memory and policy. Collaborative approaches that involve local knowledge and respect modern land use rights both improve science and honor present communities. Listen again to the quiet rearrangement. Grasses retreating, reed beds thinning, herds wandering further. Ecosystems do not flip instantaneously, but they reconfigure in ways that matter deeply to daily life. For households plotting caloric economies, the adjustments are granular and continuous. Harvest a little earlier, store a bit more, widen exchange ties, try a new net design. These ordinary adaptations are the engines of resilience. In the next part, we will look at technology and social innovation, new tools, modified shelters, and emergent practices that helped people live through colder years and build different lives afterward. Dim the lights and breathe slowly. Imagine hands, patient and precise, shaping wood and stone by a hearth. Small inventions, the hafting of a blade, a deeper pit for storage, a finer grinding slab, rearrange daily life in ways that add up over seasons. In this part, we look at the technologies and social reorganizations that helped people cope with the changing climate. Tools that smoothed effort, built-ins that protected surplus, and routines that remade cooperation. Start with craft-scale adjustments. One of the clearest archaeological signals of intensification is the proliferation and refinement of groundstone equipment. Grinding slabs, querns, and mortars become more numerous and sometimes larger, indicating a sustained investment in processing plant foods. The ergonomics change too. Deeper, more stable surfaces reduce the labor per unit of flour produced. That matters when the difference between a thin porridge and a nourishing loaf is hours of grinding each day. Small mechanical improvements, rounded pestles that reduce hand strain, or better balanced saddle querns, multiply through households as saved labor and improved nutrition. Sickle technology also adapts. Composite sickles, with small microliths inset into wooden handles and bound with plant fibers, permit more efficient harvesting. The characteristic glossy polish on microlith edges, sickle gloss, tells us these tools were used repeatedly on silica-rich grasses. Innovations in hafting techniques, repairs, and shared maintenance rituals keep tools in service longer, lowering the cost of intensive harvesting. When more people can harvest more efficiently, the local return rate for plant gathering improves, and storing becomes a more attractive strategy. Storage innovations are central. Simple pits evolve into lined or sealed features that protect seed from moisture, pests, and fungal rot. Some sites show clay-lined bins or raised ventilated containers that keep grain dry. Others use thickly plastered vessels stored in cool, dark rooms. The architecture of storage alters social time. Stored food decouples consumption from immediate harvests, enabling planning, redistribution, and the possibility of seasonal craft specialization. Secure storage also raises new social questions. Who controls access? How are surpluses shared or taxed? And what social institutions govern redistribution in bad years? Archaeology sometimes hints at answers: communal storage rooms, repeated communal repairs to granaries, and specialized spaces that imply formalized access rules. Shelter and microclimate adaptations emerge in building choices. Houses deepen hearth designs to retain warmth longer. Low doorways and wind-blocking walls reduce heat loss. Insulation from thatch, hides, or thicker packed floors improves comfort. In some sites, communal buildings, longer, more robust structures, appear or are repurposed, suggesting a pooling of labor and resources to weather harsh seasons together. These architectural changes are not merely comforts. They reduce energetic demands, protect stored foods from damp, and create shared spaces for cooperation and transmission of knowledge. Seed care practices, repair techniques, and ritual acts that reinforce social bonds. Food technology develops beyond tools into processing knowledge. Techniques for parching, drying, and fermenting increase shelf life. Smoking fish and meat, parching grains to make them less attractive to pests, or fermenting seed pastes to increase caloric extraction all extend usable calories beyond the growing season. Such techniques are labor-intensive but yield predictable returns in scarcity. They also create specialties. Households or individuals who become known for particular preservation skills may gain social roles, trade leverage, or ritual prominence. Water management and micro-irrigation appear where hydrological opportunity allows. Small diversion channels, rudimentary terraces, and simple check dams concentrate runoff and extend moisture availability into late seasons. These interventions can create local micro-refugia for cereals and wetland plants, boosting yields without large engineering projects. They reflect careful ecological knowledge, the right slope to slow runoff, the right stone placement to prevent erosion, and the right planting patch that benefits from seepage. Such practices often require coordinated labor and trust, nudging social organization toward cooperative arrangements. Mobility and transport technologies adapt as well. Improved basketry for carrying heavier loads, more efficient pack frames, and communal sledges or rollers for moving large storage containers change the logistics of redistribution. These technologies reduce the friction of exchange and make seasonal aggregation more feasible. People can move surplus to safer storage centers, and regional networks for redistribution become more viable. Social institutions evolve alongside material change. Storage and redistribution often require norms, rules for who draws on reserves, obligations for contribution, and protocols for conflict resolution. We see archaeological indicators, standardized storage features suggesting shared norms, more elaborate communal architecture that implies organized labor, and burial practices signaling lineage claims that could anchor claims to stored resources. Ritual and symbolic behaviors, depositing particular objects in storage pits, marking granaries with tokens, or involving ritual specialists in sealing ceremonies, can bind social expectation to material practice. These acts both protect resources and reinforce the social contract necessary for collective survival. Knowledge transmission intensifies. Apprenticeship in craft and food processing becomes more formalized, whether implicitly, by watching and doing, or explicitly, through instruction and deliberate training. This cultural transmission reduces risk by distributing skill. A community where many know how to repair sickles, treat seed against pests, or build clay-lined bins is less vulnerable than one where such skills are concentrated in a few hands. Innovation is not uniformly technical, it is also organizational. Households aggregate into cooperative units for large tasks, harvests, storage maintenance, or protecting herds. Exchange networks reconfigure. Instead of sporadic long-distance trade, we see more frequent, smaller exchanges that knit communities into safety webs. Reciprocal labor arrangements, sweeps of people helping to harvest or repair, and ritualized sharing create predictability. Predictability is a resource in itself, yet these technological and social gains carry trade-offs. Secure storage can create inequality, as households with larger reserves may exert greater social control. Intensified processing increases exposure to certain health risks. Gritty, ground meals can increase dental wear and respiratory exposure to smoke from longer or more intense hearth fires. Reliance on a narrower suite of resources can increase vulnerability if those resources fail. Thus, technological innovation is a double-edged sword. It buys resilience, but introduces new dependencies and social dynamics. An ethical reflection is important. Many of the technologies and practices we study intersect with living traditions, basketry styles, seed-saving techniques, and knowledge about local water management. Researchers should approach these continuities with care, collaborating with local communities, respecting knowledge holders, and avoiding extractive narratives that present ancient innovations as disembodied facts rather than cultural continuities. Picture, now, the small daily rituals of technology and care: a basket mended at dusk, a seed inspected by candlelight, a communal shove to smooth a plastered bin. These ordinary acts accumulate into different lives, new rhythms, networks, and institutions that help people persist and sometimes flourish after hard years. In the next part, we will offer an ethical note about sampling, landscape stewardship, and the responsibilities of researchers when working on places important to living communities. Dim the lights and breathe slowly. Before we move on to synthesis and closing, let us sit with an imperative that shapes every hour of careful research. The past we study belongs in many senses to the living present. Stones and seeds, bones and soil, these are not merely data points. They are fragments of landscapes that communities still care for, of ancestors who may have descendants today, and of ecosystems that remain home to living peoples. Ethical practice is not an add-on to good science. It is the scaffolding that makes trustworthy research possible. Start with consent and consultation. When scientists plan excavations or sampling campaigns, the first step must be dialogue with local stakeholders, landowners, municipal authorities, cultural heritage agencies, and, where relevant, descendant communities and indigenous custodians. That dialogue should be meaningful, not perfunctory. It means sharing research goals openly, listening to local concerns, explaining what materials will be sampled and how, and clarifying how results will be communicated. Consultation shapes sampling choices, what to sample, how much to take, and whether an alternative, non-destructive method might suffice. It also builds relationships of trust that make long-term stewardship and collaboration possible. Destructive sampling requires particular care. High-resolution radiocarbon dates, aDNA, youth assays, and some proteomics methods demand removing tiny but irreversible pieces of material from artifacts, bones, or speleothems. Ethical practice minimizes harm, choosing the smallest viable sample size, targeting already broken fragments when possible, and documenting and archiving what is removed. Labs now use microdrilling, laser sampling, and other techniques that preserve most of the object. The decision to sample should be justified in writing, shared with local partners, and, when appropriate, approved by a heritage authority. Science gains credibility when it is explicit about what was taken and why. Transparency matters. Researchers should publish or otherwise make available clear records of sampling locations, volumes, and pre-treatment protocols. This transparency enables replication, allows future analysts to assess prior choices, and helps local stakeholders understand how the research was done. Where possible, results and raw data, calibrated ages, sequence reads, isotope values should be made accessible in secure, respectful repositories. Open data accelerates science but must balance openness with privacy and cultural sensitivities. Sensitive human-related data, including genetic information, often require controlled access agreements developed with community input. Benefit sharing is not merely compensation. It is a principle of reciprocity. Communities that host research should derive tangible benefits, capacity building, training opportunities for local students and technicians, co-authorships where appropriate, and meaningful involvement in how findings are interpreted and presented. Educational outreach, public talks, museum displays, and bilingual or locally tailored materials can make research visible and useful to people who live in or care for the studied landscapes. Shared stewardship of archives and co-curation of exhibits invite communities into the ongoing story, rather than treating them as passive providers of samples. Repatriation and respectful curation are essential when human remains or sacred objects are involved. Different legal and cultural frameworks govern repatriation in different countries, but the ethical horizon is clear. Researchers must honor the wishes of descendant communities and official custodians. Where repatriation is requested, scientists should facilitate returning remains or artifacts and support culturally appropriate reburial or care. When repatriation is not requested, collaborative curation, shared decision-making about storage, access, and display respects local values and helps avoid extractive relationships. Non-destructive alternatives deserve serious consideration. Advances in imaging, spectroscopy, and remote sensing often allow important questions to be addressed without sampling. High-resolution photography, micro-CT scanning, and surface proteomics can yield insight into structure, residues, or internal composition. When non-destructive or minimally destructive options answer the research question adequately, they should be preferred as a default. Destructive sampling should remain a last resort, justified by clear scientific need and community consent. Publication ethics require care in language and framing. Scientists should avoid sensationalist headlines that overreach the data. Nuanced findings, those marked by uncertainty or regional variability, should be communicated as such, not simplified into deterministic narratives. This restraint protects the dignity of studied peoples and prevents the misappropriation of results into political or cultural agendas. It also maintains public trust, which is fragile and easily eroded by overstated claims. Privacy and respectful handling of genetic data are particular concerns. aDNA can reveal ancestry, kinship, and sometimes information that has social or political sensitivity. Researchers must develop data sharing agreements that protect individual and community privacy, define who may access genetic data sets, and outline permissible research questions. In many cases, communities may request restrictions on analyses or public dissemination, and ethical practice honors those requests, rather than treating data as free for all. Long-term stewardship of samples and archives matters. Material deposited in institutional collections should be cataloged, preserved under appropriate environmental conditions, and made available for reanalysis under agreed protocols. Where possible, physical repositories should be accessible in the region of origin. Shipping samples across borders raises legal and ethical complexities. When repatriation is not immediately possible, stable, locally accessible archives and digital surrogates provide alternatives that respect origin communities. Legal frameworks and international conventions overlay ethical considerations. Researchers must comply with national antiquities laws, export controls, and international agreements. But legality does not absolve ethical deliberation. A project can be technically legal and still ethically wanting. Ethical practice often goes beyond compliance, striving for generosity, partnership, and cultural humility. Capacity building is an active form of ethical practice. Training local archaeologists, lab technicians, and students ensures that knowledge and technical ability reside within the regions studied. Collaborative research designs that include local co-investigators and invest in lab infrastructure leave a lasting legacy beyond a single project. This is both fair and scientifically wise. Local researchers bring indispensable contextual knowledge that improves interpretation and fosters enduring stewardship. Finally, recognize that ethics improves science. Projects designed with community input, minimal sampling, and transparent reporting often yield higher quality data and more robust interpretations. When local knowledge informs sampling decisions, identifying sacred spots, suggesting where context is better preserved, or pointing to oral histories that guide interpretation, the resulting science is richer and more accurate. Ethical research practices are not constraints on curiosity. They are the conditions under which curiosity can be sustained, trusted, and reciprocated. In the next part, we will gather the threads: climate archives, archaeological signals, models, and ethics, and work toward a measured synthesis, what most scientists agree on today, and what remains open for new data to decide. Dim the lights and breathe slowly. Let us gather the threads we have laid out across ice, sea, soil, seed, and bone, and weave them into a careful, measured picture. Synthesis is not the same as certainty. It is an honest mapping of where evidence converges, where debate continues, and where the next important questions should point. As we move through this synthesis, I will separate the points that carry broad consensus from those that remain contested or speculative, so you can hold both the firm ground and the open horizon. Most researchers agree on the basic shape of the Younger Dryas as a major and abrupt climate episode centered in the late glacial interval, roughly 12,900 to 11,100 years before present. High-resolution Greenland ice cores record a sharp decline in isotopic indicators that correspond to rapid northern cooling, and that decline is followed within a millennium by an equally rapid return toward warmer conditions. Marine and terrestrial archives in many regions preserve complementary signals: colder sea surface temperatures and increased evidence of iceberg influence in North Atlantic cores, shifts from woodland toward more open vegetation in many pollen records, and changes in hydrology visible in lake sediments and speleothems. The event's timing and its north-centric magnitude are robust across multiple independent proxies, which together form the bedrock of consensus. There is broad agreement, too, that the Younger Dryas had meaningful consequences for human societies living at the time. In the Fertile Crescent and adjacent regions, archaeological evidence points to intensified processing and storage of plant foods, increased use of grinding technology, and continued investment in houses and place despite environmental pressure. Elsewhere, human responses varied: increased mobility, shifts in prey choice, and reorganization of exchange networks are common adaptive strategies visible in the record. The key takeaway is not a single uniform human outcome, but a set of regionally variable, culturally mediated responses shaped by local ecology, prior social investments, and existing networks of exchange. Human resilience and vulnerability during this interval are best read as contingent and contextual, not predetermined. Where consensus is more cautious, concerns the primary trigger or combination of triggers that initiated and sustained the Younger Dryas. Many lines of evidence are consistent with meltwater forcing, large discharges of fresh water into the North Atlantic altering ocean density and thereby weakening the overturning circulation. This mechanism is physically plausible, is supported by some marine core signals that suggest freshwater pulses, and aligns with models that show AMOC sensitivity to freshening. But meltwater is not the only plausible contributor. Internal ocean atmosphere variability, volcanic clusters, and complex feedbacks involving sea ice and atmospheric circulation likely played roles too. The sensible synthesis many scientists favor is multi-causality. The climate system was in a state where multiple forces could interact, with meltwater pulses plausibly acting as a primary or amplifying driver in some scenarios, while internal variability and other forcings moderated regional expression. Chronology remains a central pivot of uncertainty. The finest archives, some ice cores and varved lake sequences, provide nearly annual resolution, while many archaeological and marine records have date uncertainties measured in decades or centuries. Where high-resolution, well-dated terrestrial archives exist near archaeological sites, convergent timing strengthens causal inferences. Where that fine synchrony is absent, robust causal claims must be more tentative. Improved regional chronologies, coupled tephrochronology, and more UCU-dated speleothems would dramatically sharpen our ability to claim direct causal links between climatic step changes and specific human behavioral shifts. Another point of healthy consensus is methodological rigor. Extraordinary claims require strict stratigraphic control, replication across independent labs and sites, and multidisciplinary cross-checking. The debate over dramatic hypotheses, like a single cosmic impact as the dominant cause, has reinforced this lesson. When scholars apply standardized sampling, independent replication, and careful stratigraphic framing, the field advances. Where those safeguards are absent, claims remain provisional. The discipline-wide move toward transparent data sharing, replicate sampling, and conservative interpretation strengthens not only chronology, but trust between scientists and the public. Ecologically, the synthesis is that the Younger Dryas reconfigured habitats in ways that mattered to resource availability. Contraction of some woodlands, expansion of steppe in places, shrinkage of wetlands in others, and shifts in faunal communities. Those ecological shifts changed local resource economies and raised the value of buffering strategies such as storage, processing, and diversified diets. Where microrefugia persisted, river margins, terraces, and seepage-fed patches, they became anchor points for continued plant use, and sometimes for intensified harvesting that set the stage for later agricultural practices. On the social side, synthesis highlights variability and innovation. Some communities invested more in storage and processing, and thus remained in place, building incremental selective pressure on plants and social protocols for sharing and stewardship. Others adopted mobility and exchange to manage risk. Neither pathway is inherently superior. Both are adaptive responses to particular constraints. The archaeological record, when read holistically, emphasizes diversity of strategy rather than a single trajectory toward farming or collapse. Finally, synthesis must carry an ethical clause. The pursuit of more precise dates, richer aDNA datasets, and finer microstratigraphic sampling must be balanced with respect for heritage, minimal destructive sampling, and meaningful collaboration with local stakeholders. The best science in this field emerges from partnerships that include capacity building, transparent data stewardship, and shared interpretive frameworks. Those practices not only make the research ethically sound, but also materially improve the quality of the questions asked and the interpretations offered. As listeners and thoughtful readers, you can hold this balanced stance. The Younger Dryas is a sharp and meaningful climate episode that reshaped environments and human options in many places. And the scientific story is one of convergence across multiple archives rather than simple, single-cause narratives. Where the map still fades, causation in precise regional instances, exact timing of particular social responses, and the detailed routing of fresh water, opportunity for careful, ethically grounded research remains. In the next part, we will close gently. We will reflect on human meaning, the continuity between past and present, and how the lessons of resilience, contingency, and careful evidence can quietly shape how we think about our own moment. Dim the lights once more. Breathe slowly and feel the rhythm of an old landscape settling after a long winter. Picture a valley where ice has retreated and streams edge back toward their channels. Small hands clear hearths, mend baskets, and test seeds by touch. That ordinary afterwork, the careful tending of shelter, seed, and story, is as important as any dramatic headline. It is here, in the quiet accumulation of small acts, that futures are made. Across the places we have visited, a few ideas stand out in gentle clarity. First, abrupt climate does not erase people's capacities to respond. It reshapes the options available, and often it rewards ingenuity, cooperation, and the steady application of ordinary techniques. Better storage, slightly improved harvest tools, the choice to widen exchange networks, and the decision to protect specific wetland patches. Those actions do not always show as grand revolutions in the archaeology. Quite the opposite. They are often incremental, domestic, and social. But over decades and generations, they alter demographics, diets, and landscapes in durable ways. Second, the Younger Dryas reminds us that the Earth system can change fast, and that those changes have ripple effects through ocean currents, rainfall patterns, plant communities, and human systems. The physical archives, ice, marine mud, lake varves, and speleothems, preserve those ripples in different tongues. Our job as listeners and interpreters is to learn enough of those tongues to read a coherent account without forcing a tidy story onto a complex past. Where multiple archives speak in concert, we can be confident. Where they disagree, we are invited to probe deeper and to welcome nuance. Third, human responses were varied and creative. Some communities doubled down on place, refining processing and storage to smooth seasonal variation and to make seed into time. Others increased mobility, following herds or moving toward better water. Still others wove denser exchange networks, moving goods and knowledge across surprising distances. None of these patterns is inherently better. Each is an adaptive response to particular constraints and opportunities. That variability is the great lesson. Resilience often looks less like uniform success and more like a world of locally sensible experiments. Fourth, the scientific process that reconstructs these past lives is itself a human story of gradual refinement. Bold hypotheses, such as dramatic impact claims, spur useful tests and improve methods even when they do not withstand scrutiny. Progress comes from the slow layering of reliable measurements, the replication of results across independent labs, the honest calling out of uncertainty, and the ethical practices that respect both the physical archives and the living people who care for those places today. Good science is conversational. A paper speaks, others test and reply, and the narrative matures through scrutiny. There are practical echoes for our own time. The past shows that social networks, diversified resource bases, and institutions for sharing and storage increase resilience. It also shows that abrupt system-level changes can have long tails, effects that last generations, and that recovery often involves new social forms, not a return to the exact old patterns. These are not simple prescriptions for modern policy. The scale and machinery of contemporary change are different. But the past does invite humility, planning, and attention to the small cumulative acts that support survival and dignity. Before we close, a few modest suggestions for how to carry this episode with you. If you found the slow, evidence-first storytelling useful, consider subscribing and leaving a small note about when you usually listen, late nights, early mornings, or during quiet commutes. That helps shape future episodes. I place timestamps in the description so you can re-listen to specific method sections or scenes when you like. If you're curious and want to read further, look for accessible summaries from reputable journals and public-facing museum releases that emphasize methods and uncertainty rather than dramatic claims. A brief ethical reminder: the stories we reconstruct come from places that remain meaningful to living people. If you share or repurpose any of this material, do so with care and attribution. Scholarship is not just for scholars. It is part of a broader civic conversation about heritage, stewardship, and how we narrate common pasts. And finally, a personal note of quiet gratitude. Thank you for traveling in time with patience and curiosity. The work of reconstructing deep pasts is collective. Scientists in labs and field teams, local stewards and community partners, students learning methods, and listeners like you who value a careful, calm voice. Each of us carries a small piece of responsibility for how these stories are told and used. Dim the lights one last time if you like, and breathe in slowly. Imagine the courtyard we began in. Seeds tucked under cloth, hands that have learned the rhythms of care. That courtyard is both a real place and an emblem. Ordinary people responding to extraordinary circumstances with steady human practices. The story of the Younger Dryas is, in the end, a story about those practices and the slow accumulation of human decisions that shape futures. If you want more episodes like this, calm, evidence-driven, and respectful of uncertainty, I will be glad to keep telling them. When you listen, you help sustain a careful approach to history. Stay curious, stay calm.